Good afternoon, everyone. So you had one session yesterday also. Did you people attend that session? Okay. The session which was there yesterday, you have some topics left in that. So you'll have another session tomorrow, same time, same place. So for those of you who wish to attend the rest of that also, you can do that tomorrow again, 1 p.m. That's going to be the time, right? So my name is Dr. Jasmine Walia, and I will be taking a few sections of this previous year questions of UPSC Science and Technology part. So there is this document which is available on the Vision's website. It's a compilation of the last 7 years. So we are taking 2017 to 2023. So before we go into the discussion, how many of you will be writing the exam this time? Anyone? Okay. So how do you feel about the exam? How's the Josh? I should be asking you that. Okay. And the rest of you, 25? Koi 25 se baad wala bhi hai? Okay. No. All right. So see, for science and tech, the important thing to understand is that the concept here becomes very important, and irrespective of your background, because in science there is this problem also that people come from different backgrounds. Some who are from science backgrounds, they might find themselves comfortable in some sections. Again, not all sections, because science is also now going to be on very different topics. The basic premises being physics, chemistry, biology, and then of course applications of that. So if you are from a science background or from a non-science background, irrespective of that, you'll have to go back to the basics. That is one point which is very important. As we see the different kind of questions which are here, you'll understand that the questions are expecting you to really know the concept. You really need to have an idea about how things are working, and not just the facts. Factual emphasis in these sections used to be there, I mean, very prominent factual influence used to be there, but maybe, you know, that was 15 years or beyond that ago, okay? Now the current trend is more on application, the current trend is more on understanding of concepts, and that's how we'll see as we go through the different questions. Okay, so the portion which I am going to take is the basic science part, and these are the topics which are there in the basic science questions. See, as such, if you look at UPSC's syllabus, if you know, we open what UPSC has written, so they have simply mentioned science in 2 words. So they write general science, right? But you know, if you just ask somebody, go and study general science, this can be something which is endless. So previous year questions tell us that general science mein what all topics can be there. Okay, so that division of kaun sa topic padna, and again further, how much we need to study one topic, that idea we are going to get from current affairs. The syllabus is really not helpful in that sense. So we have divided this into 2 broad categories: science and technology and basic science. So basic science mein you have cell biology, then you have microorganisms, then diseases based on these microorganisms. These topics are all linked in a way. Then from diseases, how your body is fighting diseases, that will be your immune system. Then hereditary and evolution. Then properties of matter. and applications in everyday life. Different products which you use in everyday life, what is the chemical concept behind them, what are the physical properties, all of those things, they are going to be in this part. And then space science. Basic working of the universe, the planets, the stars, how all of that happens, that's going to be basic science. In our this session today, we are also going to take two parts from the science and technology part. So from technology, you have one part which I'm going to be taking is biotechnology, and the second part is going to be health related development. Okay. Again, of course, you know that UPSC question paper is not divided into these segments. We have just categorized them because then we know how we can prepare. Okay, how we go about doing that part. Okay, sources. Whenever there is a discussion on preparation of any portion, there is always this discussion on sources. We'll go through different questions, and individually also I'll be emphasizing that how a particular source can be helpful here. Okay, but on a broader level, to start this off, we have the sources which this document has labeled as essential sources, and you have essential material, essential newspaper. So essential material is going to be something which is going to be your basic point, the starting foundational point. So you have NCERT books from 6th to 10th. And for some extended concepts, like in biotechnology, class 11th and 12th. Okay. In this, I always recommend this to everyone, that irrespective of your background, whatever is your background, a basic reading of 6th to 8th will always be helpful. Even if questions are not asked directly from there, but there will be ideas which will help you solve those questions. Things like, you know, there's a question on boiling point. So you read about boiling point in these basic books. So you have to do this. And this should be your starting. In terms of, you know, our understanding of questions, also you have to visualize it like this, that you cannot solve questions if you have not studied the base. And maybe every time solving a question might not really be what you need, but an idea about what kind of questions can be your first step if you choose to look at the questions first. And then from there, studying basic foundation and then trying to solve the questions. That's when you'll get the answer. That's when things will make more sense to you. Okay, so this is definitely the starting point here. The other aspect is going to be the current affairs. Okay, so newspaper here becomes very important. Hindu, Indian Express, whatever newspaper you wish to read. But the important thing is you must have one newspaper. And I think this is also a blanket rule which any session, any discussion on preparation you go to, this is what is going to be told to you. Okay, newspaper reading is must. Because newspaper gives you a very wide base, it gives you a very extensive range of topics and ideas to think about. I've also included some cuttings from newspapers so as to understand that how we can emphasize or what we need to prepare from the science perspective. Because in science, the challenge is this also, that how much depth do you need to go to? What is the exam asking you to, especially with prelims? Because in prelims, there is a scope of asking things over a very, very wide range. They can ask you anything with 4 statements that can be put across. So when you are reading the newspaper, how much depth do you need to go into? That idea is again very important. Other things which you call reference material. Now of course, see, you have to remember that you have limited time, right? So everybody might not need the same reference in everywhere. Maybe there's somebody who understands a lot of concepts in physics, so maybe they don't have to go for extra research. For another person for whom physics is you know, something which does not make sense to them, for that they will need some extra research. These are some good sources which can be used. You have Britannica, you have NASA, ISRO, and ScienceDirect.com. Science Direct explains a lot of concept. You can just go to the website, search it. They'll have some good diagrams and explain the things. And for space, NASA and ISRO website, again, depending on what development you're looking for, they will be helpful. Other than this also, please understand this thing, कि आपने एक बार basic पढ़ लिया, then you go to the newspaper. Now, to read the newspaper or to read current affairs as such, right? Like you will have current affairs documents also. So to read that, you have to be keeping this thing in mind that you should understand that. अब वो अंडरस्टैंडिंग आपको एनसीईआरटी से आ रही है तो ठीक है, इनमें से किसी सोर्स में से आ रही है तो ठीक है, एंड अदरवाइज इवन इफ दिस इज नॉट वर्किंग फॉर यू, यू हैव टू गो at some other location to understand that, that's also fine. But at the end of the day, the objective will be that you understand what the current affairs topic is about, what the newspaper is talking about. Those terms should make sense to you. Otherwise, that whole idea of the rote learning concept, that doesn't really work here, right? If you understand, then you'll be able to solve the question. Otherwise, it'll be tricky. Okay. So basic strategy has to be NCERTs, newspapers, current affairs with proper understanding. That's the simplified idea for science and technology, and to be able to solve questions in this section. The number of questions here, you have starting with 2017, 8, then 13, 14, 13, 12, 14, 8 questions again last year. This year we'll see how many questions are there. But roughly we are aiming at somewhere around 10 questions. Okay. And these 10 questions are very helpful for you to getting that edge. Because again, like it's always discussed that there are some sections which everybody understands, right? Like polity and history. Okay, which most people will be writing, will be appearing, attempting the questions. So there the scope of you getting more marks, ultimately this is the competitive exam, right? You need to do better than the next person, and maybe not very better, because ultimately there is a cutoff also. Do you know what was the cutoff last time? Yeah, around 75 marks, right? A little over 75 for general category. So you need to get more marks than the cutoff, and you need to have a comfortable range also, because suppose the cutoff was 75. So somebody who calculated their score and had say 80 marks, that person would have been very underconfident कि अब मेंस की तैयारी करनी है नहीं करनी, cutoff कितना जाएगा नहीं जाएगा. But if the cutoff is 75 and you have 95 marks, then you are like very confident that okay, I'm going to get through, and then you don't waste any time, and then you start preparing for mains. So one confidence building exercise would be to get those extra marks, which is ultimately the game of 4 or 5 questions only. And these sections, which a lot of people don't work on or don't work very well on, they can really be that. If you analyze them well, if you understand them, then irrespective of your background, you will be able to make that difference of those 4 questions or 5 questions, which really at the end of the day can be the game changer for you, right? So there are topics here which are of high prominence, topics like health and biotechnology and space, which are more prominent, in which there are more number of questions, and which you will see them coming across in the paper more. In division between basic science and science and technology, so the total questions of 7 years is 82 questions, and out of that 21 is basic science and 61 questions is in science and technology. Further, if I have to do an analysis of this trend, specifically in different areas, like the first slide I showed you with the topics which we'll be covering, like in basic science, agar 21 questions hain, so out of this, the maximum number of questions are from space science. Space science, there is also space technology, so usme bhi questions honge, similar concepts pe, the number will become even more, okay? But 21 में से 6 तो हैं ही. So 7 years का paper है, that means at least 1 question is coming on space science every year, right? And then you have everyday applications, hereditary, immune system, diseases. Diseases also a very important area, and because इसको भी different sections में divide किया, disease, then health, then this number will also further go up. Technology में the two sections which we are going to be discussing here, you can see biotechnology 16 questions. Out of 61, 16 questions, and health 2 questions. So 16 means 2 questions on an average every year, right? There are a lot of new developments happening in this area. This is a very important part for your mains portion also. There is one fixed question at least from biotechnology every year in mains. So here, whatever new developments are happening, the concept behind that or how that is working, they are going to ask in some way. So you have to keep a lot of tab on this section. You can see ये सारे technology के parts में से you have IT as being very prominent, information technology, and even a little more than that is going to be biotechnology. And if you add basic concepts of heredity also, which is again linked to this, then furthermore the number becomes more than 16 also. So very, very important. This should also tell you that you like the subject or you don't like the subject. You have to clear the exam ultimately, right? That's your objective. So padna padega. There is no choice here, right? Going further to our discussions of the questions, how I would like to do this is, we will definitely understand what is the answer to a particular question, and along with that, an important idea is going to be how do we reach that question in the exam? Because ultimately, we all know that we are not getting the same question again, no? So, we can get themes which are same, we can get things which are similar, but might not be same. So, we will focus on why a question is asked, we'll focus on how do you get the answer to that question, and for devising a preparation strategy. If you have to study this in future at any point of time, what can be done. The first topic is going to be cell biology. Now, cell is the most fundamental unit of life. Every organism, its structure, its body, that is made up of cells. And you need to have an idea about these basic type of cells. The basic cells, the basic organisms. This is every organism, bacteria, fungus, amoeba, plants, animals, mosquitoes, they are all made up of cells. So understanding the basic structure of the cell, that is very important. That's the starting point. And you get this in your NCRTs. Okay. So 9th standard NCRT has a chapter on cell, which is going to be very, very important. Okay, so that you must prepare everything that it is talking about, different parts of the cell, all of that you should know. After understanding the basic structure, you need to know about the different types of cells. And types of cells actually are indicating the types of different organisms. So you need to have a basic idea about the different organisms which are there. So you have bacteria. Okay. What is special, what is unique about bacteria, fungi, animals, plants. These are very, very fundamental concepts in biology which you should know. Along with these organisms, you also have another category, though we cannot qualify it exactly as cells, but as a special group, you also need to know about the virus. So I'll write the virus specifically. Virus has some unique properties, we'll talk about that, because of which we cannot call it the cell. But in terms of working of different organisms, what is their action, what is their activity, virus would also be important. So ये basic layout है, ये prepare करना ही करना है. There are questions which are asked, and this is the most fundamental thing, so there will be questions which will be asked here. Taking you to the question now. So this is a 2020 question on plants and animal cells. Very fundamental question, basic NCRTs help you in this, in solving such questions. You don't really need to go into any source. इसमें कोई application नहीं है, कोई current affairs कुछ नहीं है, direct question है. The question is asking you which of the following statements are correct regarding general differences between plants and animal cells. Now plants and animals, they both have cells, but there is a slight difference between their cells. The difference is in this form. Animal cells, they have a cell membrane. Cell membrane is basically the outer covering of the cell. And plant cells, in addition to the membrane, have a cell wall. So cell membrane तो होता ही है, ये inner layer, this is cell membrane, but this outer layer, this is for extra protection, which is there in plants. And this cell wall, this is made up of cellulose. Other than this, the feature which is very peculiar for plants is the vacuole. This large yellow structure, this is the vacuole. This is basically a storage structure. For storage of things within the cell, you have a vacuole. In plants, there is only one single vacuole, but it is very large. It takes up most of the space in the cell. In animals, you have many vacuoles, but they are very, very small in size. Function is same. This is also for storage, that's also for storage, but the way it's structured, that is different. That's another point of difference. After that, you have the nucleus of plant cells pushed to one side. The nucleus of the animal cell is in the center, but here plant cell mein there is no scope for the nucleus to be in the center because the whole space has been taken up by the vacuole. So nucleus, bichara, it's pushed to one side, it's going to be in a peripheral position. Other than this, there is one more organelle called chloroplast. The name of the structure is plastids, and there are different types of plastids. They give different colors to the plants. There is one plastid called chloroplast, and this is for green color. Which parts of the plant are green? Leaves, right? So leaves will have these chloroplasts, right? Other colored parts will have other types of plastids, but this is also something unique for plant cells, not there in animal cells. So let's go to our question and see what the question is asking us. Which of the following statements are correct regarding the general difference between plants and animal cells? Plant cells have cellulose cell wall, whilst animal cells do not. Okay, so this is saying plant cells में cell wall है, animal cells में नहीं है. Right? So this should be correct. Plant cells do not have plasma membrane, unlike animal cells which do. This statement is saying कि plant cells में plasma membrane is the other name for cell membrane only. Okay. So this is saying plant cells में cell membrane नहीं होता है, animal cells में होता है. This is not correct because plant cells have cell membrane, cell wall is in addition. तो cell membrane तो होगा ही, cell wall extra है. This is incorrect. See, yeah. प्लाज्मा कोई वर्ड नहीं है यहाँ पे, ठीक है, दो वर्ड्स हैं, सेल मेम्ब्रेन, ओके, सेकेंड वर्ड इज प्लाज्मा मेम्ब्रेन, ओके, ये पूरा वर्ड है, नॉट प्लाज्मा अलोन है, एंड बोथ दीज वर्ड्स मीन द सेम थिंग, ओके. सेल मेम्ब्रेन बोलो, प्लाज्मा मेम्ब्रेन बोलो, सेम मीनिंग है, प्लांट्स में भी होता है, एनिमल्स में भी होता है, ठीक है. See now, if there is a confusion in the statement itself, like you don't understand what this statement means, or there's this term whilst which is used, what does it mean? So if you have if you understand Hindi, then this advantage you can use. If there is a confusion, read it in Hindi. The question paper has a Hindi translation of the question also. So sometimes that can come in very handy. You can read it from there and then you can understand that okay, iska ye matlab hai. ठीक है. Then mature plant cell has one large vacuole while animal cell has many small vacuoles. Okay. So first statement we know is correct. So elimination can be done at this level. Then plant cells do not have plasma membrane. This is incorrect. So 2 we have to remove and this gives us C. C, 1 and 3 is going to be the answer. Again, this term mature can sometimes be confusing, right? कि mature क्यों लिखा है? But the question is just adding this to maybe, you know, confuse you a little bit, or just to emphasize that mature here means कि fully developed है. ठीक है? The cell is fully developed, it has all its parts, that's what mature means. So, options in any case don't confuse you. There is no option as one only. Okay, so that is not really a problem here. Answer is going to be C. Going to the next question, which is going to be on microorganisms. Again, very closely related to how different types of cells work. Here, this question is about a comparison of bacteria, virus, and fungi. They are saying, consider the following. You have bacteria, fungi, virus, and which of the above can be cultured in artificial synthetic medium. Now, this term is the main thing here. Artificial synthetic medium here means a medium where there are no living components. There are no parts, no cells, nothing like that. So, probably an artificial synthetic medium is just a bunch of chemicals with some nutrients which are present in it. Now, what they're asking is, out of these three, bacteria, fungi, virus, which can grow in a medium like this? Bacteria and fungi are normal organisms, they have normal cells, they are complete organisms, but with virus, there is one special property. Virus cannot grow on its own. Virus cannot produce energy, it needs another organism, needs cells of another organism. So how virus activity works is, virus goes into the cells of another living organism, it uses their machinery, it uses their energy, then only it becomes active, then only it can reproduce, then only it can function. Without that, virus is like just any other non-living thing. So why I told you we cannot write virus in that category of cells is because virus is not a cell. Cell is a complete unit. When you say something is a cell, that means it can do all the functions on its own, everything. But virus can't do that. Virus, other cell. Virus can go in animal cell, virus can go in plant cell, virus is very small, it can go in bacterial cell, fungal cell, सब में जाएगा, उनका machinery use करेगा, then it will work. So, when a medium is artificial or synthetic, मतलब जिसमें कोई cell नहीं है, then virus cannot grow in that medium. What can grow is bacteria and fungi, there is no problem, they can fully grow. Answer is going to be A, 1 and 2 only. Now, look at the time period of this. This is 2021. This discussion on how virus is growing, how virus is being produced, this was very prominent. We're talking about the COVID-19 period, right? How was COVID-19 virus first released? Where did it come from? Wuhan province in China, right? And there are reports that there was a lab in which this was created and then it came out from there. And then WHO also did a lot of research. They went there, they wanted to see, because there are also indications which said that China ne janbujh ke nikala hai, you know, they deliberately came out with this virus, right? So can a virus be made in the lab? Yes, it can be made in the lab. Only point here is not in artificial synthetic medium. आप उसको cell दे दो, वो बन जाएगा, वो grow करेगा. Cultured here means growth. Okay, virus will grow in the lab, but not in artificial synthetic medium, in a medium which has cells. So this whole discussion on how virus would be formed, that was very prominent, and that's also the base of asking this question. ठीक है? Answer is A, 1 and 2 only. Next question is on biofilms. Now they've used this word biofilms. Basically, what is a biofilm is that some microorganisms like bacteria, they can secrete some medium. They can produce, we use the word matrix. Again, the technical logic of this is not important, but you understand the core idea. So bacteria, they are growing in a particular area, and if there is some layer, some matrix, some medium in which they are growing, they kind of get an extra protection for growing there. Like in this document, they had shown some very good images of where biofilms could be found. Like there is a pipe. In that pipe, some deposits can be there. And as material is getting stuck in those pipes, bacteria can start growing. And bacteria have that, you know, protective house or nest you can think of, where they are nicely growing. Then on a reverse osmosis membrane. Reverse osmosis is your RO systems, right? You have that water purification system. Now in that also water is there continuously. So some structures, some deposits, some impurities that can be there, and that also is like a very nice shield for the bacteria to grow. It can also happen in your own teeth. So when you have food deposits on your teeth, they kind of form what you call plaque, and in that plaque, it's then just not food, but bacteria also growing it. So a nice mixture of all those things will be there. And then in some national park also, in a water body, there can be such biofilms which are there. So the question which they have asked now. See, this is again, this was not a very prominent or very basic idea, NCERT type of idea. This is more of an application type of concept. Let's see if the options help us to get to the answer. So we are identifying the correct answer, and the statements are, biofilms can form on medical implants within human tissues. So medical implants means anything which you are putting into the body. Like say you are putting a tooth implant in your mouth. Okay, a metal tooth is supposed to be put. Same idea like you saw of plaque. Food deposit hoga, bacteria hoga, usme grow karega, protective layer hoga. That can be a very good area for biofilms. Human tissue भी है, implant भी है, human tissue है, और कुछ बाहर से आ रहा है, that means infection हो सकता है, bacteria आ सकता है. So this can happen. Then can form on food and food processing surfaces. Okay. Again, think of food items spoil because of some growth of microorganisms. So bacteria can grow on food items. If there is a food processing surface, there is an area where, suppose you know somewhere a tomato ketchup is being made, and continuously for years there is this board on which the machine chops the tomatoes. So some tomato residue is there, sticking to that surface over a period of time. फिर bacteria grow होते जा रहे हैं उसमें continuously, and biofilm can easily form. Then biofilms can exhibit antibiotic resistance. See, with bacteria, when we talk about bacteria, to kill bacteria, to destroy bacteria, the medicine which is given is antibiotic. Antibiotics are medicines given to destroy the bacteria. So when I am sick, if I have a bacterial infection, I will be given an antibiotic. That take this antibiotic and the bacteria will die. When we use this word antibiotic resistance, okay, antibiotic resistance means कि bacteria पे antibiotic नहीं काम करेगा. If you give an antibiotic to the bacteria to kill it, but it does not respond to it, then we say bacteria is antibiotic resistant. Okay. In case of biofilms, because of that, you know, protective layer, that matrix, that covering which is there, bacteria do not easily come in contact with the antibiotic. Bacteria आपने protected shield में, ठीक है? तो उतने easily, if they were like freely available bacteria, they would have died easily, but here they come in contact with the medicines with a little bit of difficulty because of that layer of biofilm. So antibiotic resistance can happen. ठीक है? अब ये जो concept है ये बहुत important है. This you will get to read very regularly in news. There is another question when we go to the health section. We'll come across this concept. There is a continuous discussion globally on antibiotic resistance. There is a question which has come on in mains also on specifically resistance concept. So ये आपको reoccurring मिल रहा है, न्यूज़पेपर में मिल रहा है, करंट अफेयर्स में सब जगह मिल रहा है, आप प्रीवियस ईयर्स क्वेश्चन करोगे उसमें भी मिलेगा. Now they have connected it to this concept. Okay, if you have just this basic sense that what exactly is a biofilm, the rest of it you can work out. All these three statements, they are correct. The answer is D. Okay. One point which I found out was that there can be some articles sometimes, like this is a clipping from an old newspaper where the heading is saying, this is just the headline I've picked up, that scientists for conserving the Kadaluni biofilm. Okay. Now, how important is this topic or how much you go into it? Without that consideration, the first point for any heading, for any headline, especially you know those dedicated science pages which you have, right? Newspaper में एक पूरा page आता है science का. Right? Usme there is always this question ki, do we need to know all about these researches which are happening in different parts of the world? Especially, you know, because a lot of those researches, their points, their conclusions are not yet confirmed. So do we really need to know about that? Even if you don't know about that, it will not be that much of a problem, but you have to try and know the word. आपने एक headline पढ़ा, उसमें लिखा है biofilm. So you need to look up what is a biofilm. Sometimes there is a subheading also, like in this particular statement, which says biofilm is the thin layer of microbes on the sediment surface of mud floods. Okay, ये एक specific example में, but otherwise it could be anywhere. Okay, so ये miss नहीं कर सकते हम. Current affairs में, newspaper में, when we are reading these terms, maybe this specific issue, yeah, where they are talking about conserving it or why they are talking about conserving it, even if you don't go into that depth, you have to be there at the first point. कि biofilm लिखा है, I need to know what is a biofilm. That's the starting point here. That will really give you a good idea about many different topics. But such a topic, again, this will not be something which will be very frequently mentioned, right? So here there is no chance of approximation in any way. If you understand what it is, then you can approximate कि हाँ ये भी हो सकता है, ये भी हो सकता है, ये भी हो सकता है. But if you don't know what it is, then this would be a question which you will have to leave conveniently and move on to the next question. We come to our next section, which is on diseases. So again, for diseases, you first need to understand the premise, or again the dimension in which you need to cover these diseases, because you can't study all the diseases and there are so many diseases which are there. So what you do is, whenever you come across a disease, first rule is prepare the disease which you are finding in the newspaper. Okay, and there is not like all generic diseases. The disease which will be common, the disease which will be important, there will be some news on that disease. Maybe the government is saying कि okay, we will have a program, or we'll have some news कि कहीं पे outbreak हो गया है, इतने case आ गए हैं, or maybe some new treatment approval is there, some form of news is there. You're reading that in the newspaper, but in that you need to know what aspects you need. Okay, so even now, right? If you have to revise diseases, you know, maybe you have your current affairs notes and now you feel ki okay, disease ka revision karna hai. There's a high chance of diseases being asked. So, kya cheezein revise karni? You can make a table also for your revision, or any way you feel comfortable. But whenever you talk about a disease, you should know the cause. That's the first point. Disease kyun ho rahi hai? If there is an organism, which organism is causing it? What is the name of that organism? Okay, if there's some other factor, then you should know that. For diseases which are spread by organisms, right, like bacterial disease, viral disease, fungal disease, there is also an idea of its mode of transmission. How is it spreading? Is it spreading through air? Is it spreading through blood? Is it spreading through soil? Is it spreading to mosquitoes? I'm sure this general idea everybody has, right? Diseases will spread through different mediums. So, कौन सा medium है? जब भी आप पढ़ रहे हो, अगर आप current affairs का भी article पढ़ रहे हो, revise कर रहे हो, make sure these 2 points are there. They are very important and this has to be known. इसके अलावा if there is a government program, if there is a national program there, then that also will be important. Maybe it's just that program which is mentioned. But if you are reading a program also for a particular disease, then also go back and look for the cause and mode of transmission. This will complete your set. This is what you must know for a disease. The trends of the question is usually around this. But for some diseases, the orientation can be slightly different depending on the importance of other things. So ये तो ज़रूर पता होना चाहिए सब diseases के लिए. National program will not be there in all cases, but if it is there, you should know. But cause and mode of transmission, very important. Other things like symptoms, diagnosis, how it should be identified, what test is there, and treatment. This you don't need in all cases. Okay, this you have to take on selectively. How again you decide this? When you come to know what the newspaper is talking about. If there is a newspaper article which is talking about the symptom of the disease only, that there is some special symptom which we are seeing for the first time, right? Like the question is on Zika. So Zika ka symptom jo hai, that is microcephaly. So when this Zika outbreak took place, it was observed that children who are born to mothers who have Zika, they have a very abnormally small head. This abnormally small head was microcephaly. So there were a lot of articles that time written about microcephaly, about the research which was going on, that why this head size is small, what is happening, how the virus is impairing the development, all of that. तो उस केस में फिर automatically आपको पता होना चाहिए कि ये तो artically इसके बारे में है, that's the theme. So I need to know that. Okay, so there you can add it, but maybe not in all diseases you need to add these things. If some new test has come or new treatment has been approved, that is in news, right? Like gene therapy treatment for sickle cell anemia. We'll talk about gene therapy, gene editing also, there are questions on that. But then you should know कि ये तो news ही treatment पे है, तो यहाँ तो treatment पता ही होना चाहिए, ठीक है? But again, you don't need to know this for all diseases, but irrespective of what is the news, you need to know the cause and how the disease is spreading. I'll take you to the question. This question on Zika came during the time period where there was a very widespread Zika outbreak in many parts of the world. Okay, so there this question was an obvious choice to be asked. So again, current affairs में जो diseases हैं, they are more likely to be asked. They are going to be the diseases which you can go and prepare. Now, for different diseases, the organism which causes the disease is called pathogen. Pathogen is the disease causing organism. So, अगर bacteria disease cause करे, तो bacteria pathogen है. अगर virus disease cause करे, तो virus pathogen. Okay. Here they are talking about Zika virus infection. So virus is the pathogen, name of the disease can be Zika. Okay. Now they are saying, they are giving two statements on how this disease is spreading. First point which you need to keep in mind is, for one particular disease, there can be more than one ways in which it can spread. ऐसा नहीं है कि एक disease सिर्फ एक ही pattern से spread होगी. There can be a disease which can spread in more than one way also, and Zika is a very good example of that kind of disease. So if you look for what is the mode of transmission of Zika, it can spread through a mosquito. The name of this mosquito is Aedes mosquito. Then it can spread through sexual transmission. When you say sexual transmission, it is the transmission of body fluids. Okay, the sexual fluids, they come in contact. That is how the virus can spread. And it can also spread from the mother to the child when the child is being born. So in that birth process, the disease can be transmitted from mother to child. So mosquito से भी है, body fluid से भी है, mother to child transmission भी है, different ways for transmission of Zika. Now the statement is saying in tropical regions, Zika virus is transmitted by the same mosquito which transmits dengue. तो अब दो चीज़ें पूछ ली उन्होंने, डेंगू का ट्रांसमिशन भी पूछ लिया, जीका का भी पूछ लिया. Zika, dengue is a disease which is like more common. Every year it is in news, every year it's happening. Which season is dengue happening? What time of the year? Rainy season, right? Why rainy season? More mosquitoes, right? Diseases which, yes, breeding of mosquitoes is more. Dengue is also spread through Aedes mosquito. Okay. As such, see, tropical regions very favorable for growth of mosquito. Okay. And monsoon season, rainy season, water logging, humidity, further very good for growth of mosquito. So diseases like these, the diseases which are caused by mosquitoes, एक word हम use करते हैं vector. Mosquito को बोलते हैं vector, that means it is a carrier of the disease. So जो vector से diseases होती हैं या mosquito से diseases होती हैं, their incidence becomes highest when there are more mosquitoes, like in the rainy seasons and in tropical regions, ideal for growth of mosquitoes. So first point is correct. Second is sexual transmission of Zika virus is possible, yes, right? So both these statements are correct. Answer is C, both 1 and 2. Okay. Again, a direct question from current affairs. This disease was in a lot of news at that point of time. The next question is on plant diseases. So question says, consider the following: birds, dust blowing, rain, wind blowing, which can spread plant diseases. Now see, when they're talking about this spreading of plant diseases, again you have to think of pathogens. Plants के लिए भी disease causing organisms होंगे pathogens. Bacteria हो सकता है, fungus हो सकता है, virus हो सकता है. All of these, and there can be a lot of worms, there can be many other organisms which can cause diseases in plants. Okay, fungal diseases is one category which is very common, but all other kinds of diseases can also happen. Okay, now they've asked a very generic question here, a very basic question, where they've asked how these pathogens can spread, basically, or how these diseases will spread. So, what do you think can be the answer here? C. Okay, you don't like rain? Can human beings have a disease through water, through rain? Yes? Water logging, growing of different pathogens, then that water, if human beings drink, they can have a disease. Similar concept can work for plants also. Plants, rain, water, ज़्यादा बैक्टीरिया ग्रो होंगे, ठीक है, या ज़्यादा फंगस ग्रो होगा मॉइस्चर के वजह से डिजीज़ हो सकती है. डस्ट एंड विंड का कॉन्सेप्ट वही है कि डस्ट एंड विंड से कैरी हो रहे हैं पैथोजेंस. जैसे फंगस है, फंगस गेट्स इनटू अ वेरी ड्राई स्टेट कॉल्ड स्पोर्स. ओके, फंगल स्पोर्स के फॉर्म में होता है, एंड दीज स्पोर्स can travel with the help of wind, with the help of dust particles blowing from one part to the other. Birds may be same thing. Birds can on their bodies, you know, they are going to different surfaces. तो एक plant पे गए, उस plant के leaf में कोई viral infection था, that gets attached to the bird, then the bird goes to the next plant, वहाँ पे deposit कर दिया, plant disease spread हो गई. ठीक है? This is a question where again using some very basic common sense you should be able to conclude the answer. And these are questions which you cannot miss. See, the analysis of previous year questions or any time when you're doing a question, this idea is very important कि कौन से question miss नहीं करने और कौन से miss करने. This strategy of how many questions you play out and what will be your accuracy, this also you need to devise along with everything that you study and prepare for. So this is a question which should not have gone unattempted by anybody who was doing. This is not related to any kind of a fact, but a very basic idea. Okay. Don't try to eliminate something if you can't find a direct connection. Most of the times, most thing will work. Okay. Because they're asking how it is spreading. If they're asking which can cause a disease, maybe there could be an elimination. कि हाँ इससे disease नहीं होगी या इस pathogen से इस organism से disease नहीं होगी. But spreading, more generic, spreading can happen.
Can you show us a picture of plants or leaves which have diseases through rain?
See, again, इसमें you also need to understand that these diseases and pathogens, they will not have a fixed relationship like vectors. ठीक है? जैसे वेक्टर में मैंने आपको बताया कि एडीज मॉस्किटो में ही जीका वायरस जाएगा. Fixed relationship है. Zika virus will not go into any other in any other mosquito. For such kind of transmission, you know, where mechanical transmission is there is no fixed rule कि यही पैथोजन जाएगा या यही पैथोजन जाएगा. आप इसको रेन से कंफ्यूज मत करके आप वाटर से सोच लो इसको. वाटर बॉर्न डिजीज.
I cannot imagine how water or rain can impure the plants and leads the disease.
Okay, so do you know about anything about fungus? Okay, फंगस की एक प्रॉपर्टी है, फंगस को मॉइस्चर बड़ा पसंद है. ठीक है? Okay. So wherever there will be moisture, wherever it will be moist, it will be warm. That is ideal for growth of fungus. ठीक है? Right? एक जो potato है ना, potato को एक बहुत common fungal disease होती है जिसको बोलते हैं blight. Okay? So blight जो है, that is a fungal disease. Again, fungus, yeah, sometimes, you know, fruits and vegetables, they can rot in your house also when they're just kept. अगर पानी में रह जाए, ठीक है? आलू को एक्सपेरिमेंट करना पानी में डुबो के रखने का, ठीक है? Okay. Next is on hepatitis. Again, hepatitis stays in news. There is a World Hepatitis Day also which is celebrated. Hepatitis, there is a very large-scale organizational work for creating awareness against hepatitis. It is the yellow ribbon movement. Like you might know that there's a red ribbon for AIDS. So hepatitis के लिए there's a yellow ribbon. So ये भी हमेशा news में रहता है. ठीक है? When again this disease is being discussed, its cause would be important. Now, for viral diseases, their vaccines will be very important. In fact, for all diseases, if you can have a vaccine, you give the vaccine before you have the disease. To prevent a disease, you give the vaccine. Viruses mein, the challenge is we don't have a very good treatment options for viral diseases. So here, the vaccines are even more important. So कौन सी vaccine है, कौन सी vaccine नहीं है, that stays in news. Like recently, India has made a hepatitis A vaccine. So again, hepatitis was in news because of that. I'll tell you a little bit about hepatitis. Hepatitis is a disease of the liver. Okay, the main organ involved here is liver, and it is mainly caused by different types of viruses. और virus का नाम है hepatitis virus. For most viral diseases, this is the case. Virus और disease का नाम same होता है. Dengue virus, dengue disease, Zika virus, Zika disease, hepatitis virus, hepatitis disease. They are of 5 types. This virus has different species or different varieties. They are all damaging your liver, but their structures are different. So we have hepatitis A, we have hepatitis B, hepatitis C, D and E. The way they are transmitted, that is also slightly different. Hepatitis A through food and water, hepatitis B through blood and body fluids, hepatitis C again, similar transmission, blood and body fluids. Hepatitis D happens to people who are already infected with hepatitis B. It cannot impact alone. अगर किसी को hepatitis B का infection है, there can be a situation where he can be further attacked by another variant, hepatitis D. And E is also going to be through food and water contamination. Okay. But these are all different viruses as their structure is different. Out of all of these, we have made vaccines for hepatitis A, for B, for D and E. Hepatitis C, as of today, has no vaccine against it. And a few years ago, the discovery of this virus, hepatitis C virus, it was discovered and that received the Nobel Prize. So again, this was in news. In terms of preparation from the point of view of the exam, see there are 3 Nobel Prizes associated with science. So you have physics, you have chemistry, and you have medicine. These 3 are a very good base for questions. Prelims or mains both have seen questions from this area. And just to broaden this a little bit, ideally, what you can do is you can prepare Nobel Prizes for the last 3 years. I mean, whichever year you choose to write the exam, that year and 2 years before that. That will give you a little bit comprehensive coverage, because once a Nobel Prize is there, if it's linked to a recent development which has taken place, there will be reoccurring news and developments on that. Okay, so chances of that coming into a question is very high. We've seen this happening very, very repeatedly. So Nobel Prizes is a theme which you cannot afford to miss. Please keep that in mind. Bringing us to our question. Which one of the following statements is not correct? Hepatitis B is transmitted much like HIV. Okay. So I think HIV transmission everybody understands, right? HIV is transmitted through blood, through body fluids, sexual fluids. So hepatitis B is also similar. Okay. Hepatitis B, unlike hepatitis C, does not have a vaccine. This statement is saying hepatitis B is not like hepatitis C. Why? Because hepatitis B does not have a vaccine against it. It's wrong. Okay. And incorrect question, incorrect statement find karna hai. So this is simply the answer. Usually in a question like this, the design is such, they'll keep a simple statement which will be incorrect, and then they will bring out some very complex statements and give it as the correct statements, so that people keep getting confused. Like look at these other two statements. You might not have read about them, you might be confused, but that kind of draws emphasis away from something which you know. हेपेटाइटिस आपने पढ़ा तो आपने ये पढ़ा ही होगा कि हेपेटाइटिस सी का वैक्सीन नहीं है. This was the only thing which was supposed to be identified, but it can be missed in the burden of these other two statements which are there to confuse. Globally, the number of people infected by hepatitis B and C viruses are several times more than those infected by HIV. That is absolutely correct. Then, some of those infected with hepatitis B and C viruses do not show the symptoms for many years. This is rare for most viral diseases. The virus can just be sitting in the body and not giving you any symptoms. It might take a lot of time before the virus can start or give a symptom. So the person might not have disease for many years. Even when you test the blood of the person, they will find the virus. So this is also true for hepatitis. The only thing which is incorrect, there can just be one answer, 1 point correct, which is that hepatitis C does not have a vaccine against it. See, again, it is very important that you read the question carefully. What I used to do is, and what I always suggest is, you please underline this, right? It's easier to miss it. ये block में भी नहीं दिया होगा, right? So आप पहले तो इसको पढ़ो. When we read questions, our tendency is we read the options very carefully. You know, option ध्यान से पढ़ना है, एक एक word. But इसको हम ignore कर देते हैं, क्योंकि वो autopilot पे है कि ये तो correct option ही find out करना है, right? That is a very, very big mistake. And you know, even if you lose 2 questions like this, that's a very, very big loss. Okay, you cannot afford to do this. So, सबसे पहले question का statement पढ़ो. What it is it asking you correct statement, incorrect? अच्छा, एक और तरह से पूछते हैं, they write which of the following, all the statements are correct except. Okay, so not correct भी नहीं है, correct, and फिर then one word in italics in the end they've written accept, which is very easy to miss. So when you read this, underline this, block, target it, and then look at the answers. ये बिल्कुल miss नहीं करना. This is a very big distractor in the exam. And you also need to keep this in mind that if there will be a negative question, if it's asking for an incorrect question, the statements will be lesser known. That is by design. कि आपको statement पढ़ के लगना चाहिए कि ये गलत है. जैसे ये पढ़ के it should look like you know ऐसे कैसे कि virus है और disease नहीं है, symptoms नहीं है, right? It might be that trigger. That is again by design. So you have to be very careful and very sure about what you know. कि जो पता है at least that you should be able to deliver. There will be questions which you don't know anything about, but at least here something which was as simple as this should be correct. There could be a silly mistake again, which ideally, if you read carefully, well, should not happen. And the next question, in this document, it has been categorized here. This will fit in here also. This will fit in biotechnology, genetics also, because this is based on the working of genetic material of viruses. Okay. I'll tell you a little bit about how genetic material works. See, every organism contains genetic material or DNA. Okay, you heard about DNA? Yes? Okay, even if you have not, okay, let me tell you this first then. Before going into biotechnology and applications of, you know, say genetically modified plants and recombinant technology and all of that, you need to have the basic fundamentals of genetics on point. You need to know what is genetic material. The main genetic material which you have is DNA and RNA. You need to know what they are made up of. Okay? What is their structure? Not exact like chemistry formula, not like that, but again, what all things it is made up of, how it works. The role of this genetic material is to carry information for working of an organism. Like the most simple understanding is that what kind of hair I have, what kind of voice I'll have, what kind of nails I'll have, how my liver will work, how my pancreas will work, what kind of diseases I will have. All of that information is there in my DNA. And how am I getting this DNA? How does it come to my body? It comes from my parents. That's why, you know, my hair can be like that of my father. My height can be like that of my mother. Okay, I can have diabetes, which my mother and grandmother and her mother, everybody in the family had. How this information is transmitted is through DNA. Okay, so for every organism, from the smallest organism like say bacteria, fungus, even virus, every organism has genetic material, DNA or RNA, which works for determining all its features. So, here we are talking about the genetic material and the working of viruses in particular. When we talk about living organisms, all the organisms which we have, they all have DNA. Okay, bacteria, fungus, amoeba, crocodile, cow, monkey, सब में DNA ही होता है. Viruses जो हैं, when we talk about them and when we look at their working in terms of genetic material, there are viruses which can have DNA and there are also viruses which can have RNA. When we say viruses have RNA as their genetic material, it means their genetic information of how this virus will work, how it will attack the cell, how it will produce more viruses. That whole information is in the form of RNA and not DNA. Other than virus, this is not the case for any other organism. Like we also have RNA in our body. It's not that we don't have RNA, but उस RNA में हमारी main information नहीं. Our main information is in DNA. This is for all organisms with the exception of virus. Now, how this DNA and RNA system actually works? Okay. These are the basic structures of how DNA and RNA works. This is a DNA. So think of this first as a cell of your body. You have DNA and DNA has information for all your features. How this DNA's information will work is when some protein is produced in the body. जैसे suppose I am saying कि DNA के पास information है to make your hair color. What should be the color of your hair? That information is say in your DNA. Your DNA decides that. How that works is DNA से that information goes to RNA. So DNA creates a piece of RNA with the same information and then RNA helps in production of proteins. ये जो protein बनेगा ultimately, that decides your hair color. Okay. This functioning of DNA and RNA, this you will have to study. Okay. This is there in your class 10th NCERT also, and then a part of this is going to be there in your 12th NCERT also, advanced formulation. 10th वाला भी आपको पढ़ना है और further understanding के लिए जहाँ भी requirement है, you will have to read the 12th standard NCERT also. 12th is more advanced, 12th is more complex. So you can't read it like cover to cover कि chapter के first word से last word तक. You can't read it like that, but read small paragraphs, read topics which you are looking for. Like if you're looking for structure of the DNA, just read that topic on structure of the DNA. Okay. So DNA से RNA बनता है, RNA से proteins बनता है, ये normal system है. When proteins are formed, then that hair color actually comes out, your eye color comes out, or a hormone comes out, or whatever needs to be done. Now viruses, what they are doing, if a virus has DNA, तो DNA से वही pattern होगा, DNA से RNA बनेगा, from RNA proteins. Okay. If it is an RNA virus, there are two categories here. Further, one is called non-retro. In this, because it has RNA, from RNA straight away it will form proteins. And there's another category called retrovirus. This word retro means going backwards. Okay, going backwards. How this is going backwards is, this is an RNA virus, so it has RNA, but instead of making proteins, it makes DNA. उल्टा जा रहा है, वापस जा रहा है. Like retro music or retro fashion would be, say, fashion trends of the 80s. Okay, so this virus is also going backwards. The DNA से RNA बनता है, but RNA से यहाँ DNA बन रहा है. So this is a retrovirus. And again, see, because of COVID, all this was very highlighted. All this was in news. That how this whole thing is working, different types of viruses, they're attacked. So that's one base when the questions, there were a lot of questions asked on this idea of virus itself. Okay, so this is one of the questions. DNA virus ka example, if I have to give you example here. So you have a virus variety called adenovirus. Adenovirus is a DNA virus, a very common virus, cold, eye flu, okay, and many other kind of infections in the body can be caused by this virus. And इसी virus के बहुत सारे species हैं. They're doing different things, and there are some of these virus varieties which actually don't do any damage. Your body doesn't recognize them also, no attack, nothing. ऐसे भी adenoviruses होते हैं. Another example can be chickenpox virus. Your non-retro kind of RNA virus, this is where your COVID-19 came. Then dengue virus. And यहाँ पे retro में एक ही characteristic example है, which is HIV. The virus which causes AIDS. Now, we have terms to describe how this DNA is working. The steps which we have studied, those steps have names. So, when it is DNA to RNA, the step is called transcription. When DNA forms RNA, it's called transcription. And when RNA forms proteins, it is called translation. Okay. In case of a retrovirus, where from RNA DNA was being formed, the process was called reverse transcription. Transcription है DNA से RNA, अगर RNA से DNA बन जाए, reverse transcription. Okay. And when cells create new cells, every cell has DNA. So every new cell will also need DNA. For that, DNA also replicates. Replication means DNA also gets copied. एक DNA से एक DNA अपनी ही copy बना लेगा. It will copy itself so that it can go to new cells. So you have DNA replication, transcription, translation. And as of now, we have technology to do this whole thing in the lab also. We can make copies of DNA in the lab. We can make RNA artificially in the lab. We can perform this whole exercise, create proteins in the lab. We have technology to do all of this. So that's why we can explore applications of this. We can create technology based on this. The question here is just about examples. Yes.
And dsRNA.
Okay. So the question which has been asked is, there are types, different types of RNA. What about that? See, the thing about RNA is, RNA, if I go by what it is, its full form is ribose nucleic acid. Okay. In this, RNA is basically a small piece of genetic material. Okay, and this RNA can be doing different functions in the body. Like DNA ka function, which I told you, one main function was it holds your main genetic information. It holds how hair color, eye color, all of those features will be decided. DNA ka role fixed hai. But RNA can play many different roles in your body. Based on what role it is playing, we can have different types of RNA. Like we have mRNA. Okay, ये जो RNA था, जो DNA से information लेकर गया to make proteins, this is acting as a messenger. Because this is acting as a messenger to take DNA's information to make proteins, it's called messenger RNA. Okay, there can be many other types of RNA also, which we use in different types of technologies. Okay, so depending on where you are reading it, you will come to know that what is this RNA. The common point without being confused is कि ये राइबोज न्यूक्लिक एसिड है, इसका बेसिक स्ट्रक्चर सेम होगा, ठीक है, बट ये फंक्शन अलग अलग कर सकता है. बेस्ड ऑन दैट वी कैन हैव मेनी डिफरेंट टाइप्स ऑफ RNA. दिस इज वन RNA. वील टॉक अबाउट, लाइक यू मेंशन्ड, IRNA. सो हम IRNA की भी बात करेंगे. वी हैव अ क्वेश्चन ऑन दैट आल्सो. ठीक है, RNA कैन बी डूइंग डिफरेंट फंक्शंस इन द बॉडी. जी जी बोलिए.
Ma'am, we have read that RNA is single stranded.
यस.
Then what about the term dsRNA? What is it?
ओके, सो देर इज दिस आइडिया, जैसे इसी डायग्राम में है. इफ यू लुक एट द इमेजेस ऑफ DNA एंड RNA here, देर आर टू स्ट्रैंड्स. ये जो है DNA के एक और दो, ऐसे दो स्ट्रैंड्स हैं विच आर इंटरट्वाइंड विद ईच अदर टू मेक अ स्पाइरल स्ट्रक्चर. एंड RNA जो है, इट इज जस्ट वन स्ट्रैंड. इफ यू कैन सी हियर एंड कंपेयर, राइट. सो जो RNA होता है वो होता है सिंगल स्ट्रैंडेड, एंड DNA जो होता है वो डबल स्ट्रैंडेड है. ये एक जेनरिक कॉमन रूल है इन मोस्ट केसेस, बट देयर कैन बी एक्सेप्शंस टू दिस. There is a possibility of DNA being single stranded also. Okay, it's rare, but it can happen. There is a possibility of RNA being double stranded also. So this is applied again in most of these situations, but इसमें exceptions होंगे. How you will keep a note of it is when you are reading about one type of RNA. Okay, so there it will be mentioned. जैसे IRNA की हम बात करेंगे, RNA interference, or any other variation, there is sgRNA, there are many other varieties, वहाँ पे लिखा होगा कि double stranded है. So whenever something is an exception, and living systems में there will always be exceptions, you need to note that exception, and you also need to think about the fact that this can be a probable question also, right? Same way for DNA. There can be some situations in some viruses where the DNA can be single stranded also. It's very rare, but it can happen. So आप जो भी variety पढ़ रहे हो, if there is an exception, make sure you note that. ठीक है? Okay. Now this statement is saying adenoviruses have single-stranded DNA genomes. ये जो word genome है, this is to describe the complete genetic material of an organism. If you are talking about all the DNA of my body, the reference of that is my genome. Okay, one person's genome. If you are talking about a virus, virus में कितना genetic material है, that becomes virus's genome. Okay, और bacteria की बात हो रही है, bacteria's genome, tiger's genome. Any organisms whose complete genetic material is being described, that is the genome. So adenovirus have single-stranded DNA genomes, whereas retrovirus has double-stranded DNA genomes. Okay. Now in this question, suppose you have this confusion कि यार adenovirus में DNA single-stranded है या double-stranded है. Retrovirus should be clear. ये एक बहुत common terminology है. You should know retrovirus में DNA नहीं होगा. Retrovirus has RNA. Retrovirus is an RNA virus. So the first statement has to be incorrect. The second is saying common cold sometimes is caused by adenovirus. This is correct. Whereas AIDS is caused by a retrovirus. So HIV virus causing AIDS. This is correct. Answer is going to be B2 only. After this, we are coming to the next part, which is the immune system. We've spoken about diseases. Now we're going to see what our body is doing about those diseases. In this, vaccine part also comes. Okay, so if you include that also, then you can think of a very large number of questions, but specific working of immune system or working of any system like digestive system, respiratory system, questions are not many. Again, immune system became important with coming of COVID and a lot of discussion on how immunity is supposed to work. So this is a question. Which one of the following statements best describes B cells and T cells in the human body? So, for providing immunity, your body has cells called lymphocytes. They are a type of white blood cells and they provide immunity to the body. They are of two types. So, you have B cells and you have T cells. Lymphocytes further are of two types. Okay. So if you know this much, let's see if we can get to the question. Immune system's job is to provide protection to a person. We have different diseases and different disease causing organisms coming into our body. So our body has an inbuilt ability to fight those diseases, to be able to protect itself from those diseases, right? That's why when somebody falls sick very often, we'll say कि इसकी immunity बहुत low है, right? You know this terminology. Immunity is less, so this person keeps getting diseases. That means the body's ability to fight different diseases is lesser, so more diseases. So उसमें से let's see if we can find the answer based on this idea. They protect the body from environmental allergens. So first statement is talking about allergy. Then they alleviate the body's pain and inflammation. So they reduce pain. Then they act as immunosuppressants. Immunosuppressant would be something which is reducing immunity, okay, suppressing the immune system. And the last option is they protect the body from diseases caused by pathogens. So if you understand this very basic idea of immunity, that how it is supposed to provide protection, the answer out of these options, the option which is most appropriate is the last one. They protect the body from diseases caused by pathogens. Pathogens, I have told you, disease causing organisms, and they are protected with the help of these T cells and B cells, the lymphocytes. Okay, I understand. You might think that this allergic reaction is there, right? And allergy is also somehow related to your immune system also, only, though not with lymphocytes. ठीक है? आपको ये सही लग रहा है? ठीक है? What about this? क्या ये आपको गलत लग रहा है? Okay, okay, ठीक है. See, there'll be a lot of questions where it might seem like कि two options are correct, right? This is a very common observation कि ये भी सही लग रहा है, ये भी सही लग रहा है, ठीक है? So you start reading, right? You read from the first option, then you read the second option. You feel कि pain वाला नहीं लग रहा है, immunity कम करने वाला भी नहीं लग रहा है, ठीक है? दो हटा दिए, अभी 50% chance है. Now, the UPSC question paper on the first page says the most appropriate answer. Select the most appropriate answer. Okay. You really they don't use the word correct or anything like that, right? इन दोनों में से अब आपको जो ज़्यादा लगे वो आप choose कर सकते हो. राइट? आपके पास दो ऑप्शंस हैं, आपको दिस इज द प्रैक्टिस विच यू नीड टू डू, दैट एवरी ऑप्शन हैज टू बी ट्रीटेड ऑन इट्स मेरिट, राइट? इफ यू फील ये आंसर हो सकता है तो इसको क्रॉस मत करो, जो लग रहा है आंसर नहीं हो रहा उसको क्रॉस करते रहो साथ साथ, राइट? इफ यू स्टिल फील यू आर नॉट श्योर, लीव दिस क्वेश्चन एट द मोमेंट, यू कैन कम बैक टू इट अगेन, राइट? वेर यू फील कि आई एम कंफ्यूज, ये भी लग रहा है वो भी लग रहा है, दोबारा पढ़ो उसको बाद में, वंस यू गॉन थ्रू ऑल द क्वेश्चन, सो दैट विद अ फ्रेश माइंड, विद अ फ्रेश आउटलुक, यू कैन अंडरस्टैंड दिस. और इसमें से जो बेटर प्लेस्ड है, then that you can mark as an answer. In any case, you still have 50% chance of getting the question correct. ठीक है, दो तो eliminate हो गए. Okay. Yeah, I'm going to explain B cells and T cells. See, B lymphocytes and T lymphocytes, they are both for taking care of pathogens, the unwanted things in the body. But the way they act, that is different. Okay. ये हैं हमारे बी सेल्स जो बहुत गुस्से में हैं इस वक्त, ओके? एंड वी हैव दिस टर्म एंटीजन. एंटीजन इज द टर्म फॉर एनीथिंग विच स्टिम्युलेट्स द इम्यून सिस्टम. सो whenever something comes from outside, like a pathogen has come, ठीक है? इस केस में ये बैक्टीरिया है, ये बैक्टीरिया आए हैं बॉडी में, they want to cause a disease, and once they come, the body recognizes them as an antigen. Recognizes them as an antigen means the immune system will become stimulated, right? This is stimulated immune system, angry immune system. And these B cells, how they are going to take care of this antigen or this bacteria is, they are going to produce special proteins called antibodies. Once they produce these proteins will go and attack the bacteria, and their job is to destroy the antigen or destroy the bacteria. So B lymphocytes का काम है, whatever pathogen has come, that is going to be attacked by production of protein, special proteins called antibodies. Based on what kind of attack has taken place, for that special protein will be made. जो बैक्टीरिया उसके लिए एक टाइप का प्रोटीन, वायरस है डिफरेंट प्रोटीन, फंगस है डिफरेंट प्रोटीन. This is how B cells are providing protection. T cells are also doing this protective job, or T lymphocytes, but their mechanism is different. Aim is same. Aim is again to provide protection against different diseases, but the way they do it, that is going to be different. How they'll do it is, in this case, there's going to be a direct attack. This is a cell, this is your T cell, and this is a virus. What this T cell does is, it brings this virus inside the cell. And once it is inside, it's going to destroy the virus. So here, no formation of antibodies or anything, direct killing is happening. So T cells are also killing or destroying the pathogens. B cells are also doing that, just their mechanisms are different. Okay. They're both supposed to provide immune response. Next is your heredity and evolution. In this, you have this question of Denisovin. See, this question, again, current affairs kind of topic, and not a very common topic. But what happened was that there was this new, some fossil remains were found in some parts. It was a new discovery, and based on that fossil, they thought, or they have devised this idea कि एक special type के human beings थे पहले, early human beings, and they are called Denisovin. Of course, this research is very recent, and because of that, we don't have the complete idea about that species, like other species of human beings, right? Currently, we have only one species of human beings, right? What is the name of human species? Homo sapiens, right? So we just have one Homo sapien. So in the past, we have this idea that there was a time where there were more than one Homo species, but they all got eliminated and we just have one now. We keep getting evidences, however, from some fossil remains that there might be another species in the past. Now, again, this is the full detail of this organism is not available, and the question is also like that. ठीक है, question ने कुछ नहीं पूछा आपसे, just it wants you to know if you have read this word, if you've come across this word. So the word Denisovan is sometimes mentioned in media in reference to it is an early human species. Okay, fossils of dinosaur, of a kind of dinosaur, a cave system in northeast India, a geological period in the history of Indian subcontinent. इस क्वेश्चन में कोई अप्रोक्सीमेशन, कोई एलिमिनेशन कुछ नहीं लग सकता. You either know it or you leave it. Such questions, it can be a very smart move to leave these questions, because again the name is also not indicative of anything. कुछ भी हो सकता है. ठीक है. This, however, was again being covered in a newspaper when it was identified. Like, this is a cutting, I've taken this from the Hindu. So this is saying that Denisovans lived in Tibetan plateau, and fossil evidences have claimed that. Okay. After that, the very next year, there was this person who won a Nobel Prize also in evolutionary biology, which was explaining that there can be earlier species of humans. There was a lot of research. So when that Nobel Prize was given, again, these kind of studies got more emphasis and more reporting and more, you can say, footage. So news में था, बहुत ज़्यादा prominent नहीं था, but again, like I told you, कि at least headlines we shouldn't be missing. Okay, कि कम से कम जो headline में आप पढ़ रहे हो, look that up and keep that in mind. Okay, even if current affair magazines and other content doesn't cover it, but अगर आपने, if you open that newspaper, you made that effort, then this should be known to you. That's the idea. इस species की जो picture है, again, we still don't know the complete idea about this organism. ये इसको species का नाम भी नहीं दिया अभी. Okay, like Homo sapien है, तो ऐसा कोई Homo ऐसे करके नाम इस species का अभी नहीं दिया, because the full features of this has not been studied. We've just given this name Denisovans. They might be there, we have to construct. And there is also this species called Neanderthals, so they have some common genetic link with them. But ये species अभी define नहीं हुई. ठीक है, this has happened in 2010, so it's very early for this, right? Picture, again, a lot of people have estimated from the fossils कि इस type का jaw हो सकता है, teeth मिला है, तो you know how big the jaw should be to fit a tooth like that. Based on that approximation they have devised, but again, it's not exact. After this, we have properties of matter. Okay. This part on properties of matter, again, reading 6 to 8 NCERTs is very, very important here. It will give you, again, the concepts which you need. The questions which they ask are usually not on concepts, but on applications of concepts. So only if you understand the concept, then you can make the question. Current affairs में से भी कुछ correlation हो सकता है, but it can just be a simple conceptual question like this. Okay, ये भी एक common theme है UPSC का, that when they pick up everyday concepts to ask questions, pressure cooker, vacuum cleaner, clock, you know, such things they can ask how they work, because the idea is that you should have a general awareness on how things and systems work around you. So this is a question on a pressure cooker. In a pressure cooker, the temperature at which the cooker depends, food is cooked depends upon which of the following. Okay. So why do we use a pressure cooker? What's the point of using a pressure cooker? To make pulses. अच्छा, दाल बनाई आपने, तो open pan में बनाना versus pressure cooker में बनाना, what is the difference? जल्दी बन जाता है, right? Time taken to cook food is reduced, right? What so cooking में you need heat, ठीक है? Heat दोनों cases में मिल रही है, direct flame और pressure cooker में भी. But there is a difference in the time which is taken to cook. Why there is a difference in the time is because the food will be cooked at a higher temperature. In a pressure cooker, if you're boiling potatoes, the potatoes boil soon because the pressure cooker can provide a higher temperature within a shorter period of time, right? And how that works is by changing how water will boil. Water has a boiling point. That means the temperature at which water boils. What is the normal water boiling point of water? 100 degrees Celsius, right? Normal temperature, normal pressure conditions mein water will boil at 100 degree Celsius. Now, this boiling point is determined by what we call pressure of the atmosphere, or there is a pressure which is coming from within this water and there is a pressure जो बाहर से आ रहा है, ठीक है? When this pressure of water and the pressure from outside, when they will become equal, that is going to be the boiling point. This is dependent on pressure. If the pressure increases, boiling point will also increase. When you put this water in a pressure cooker, you are increasing pressure. Atmospheric pressure से ज़्यादा pressure है because it's going to be closed, it's going to be sealed at all ends. And instead of 100 degrees Celsius now, water will boil at a higher temperature. So that will be say 120 or more or whatever. So at that increased temperature, the cooking will become faster. Because what happens once something starts boiling is, जब water boil होता है तो होता क्या है? What is happening? So liquid is changing into gas, right? This you understand? ठीक है? तो फिर इस process को भी energy चाहिए होगी, right? जो पानी है, अब वो evaporate हो रहा है, वो boil हो रहा है, उसका gas बन रहा है, ठीक है? So a lot of energy, a lot of heat which you are giving, that will go into this, changing liquid to gas. Right? If we increase the boiling temperature, that will give us more time before this process takes place. And with that higher temperature, under higher pressure, the food is going to be cooked earlier. क्योंकि उसको टेम्परेचर ज़्यादा मिल रहा है तो वो जल्दी ब्रेक डाउन हो जाएगा, कुक हो जाएगा, सॉफ्ट हो जाएगा बेसिकली. दैट्स द होल आइडिया ऑफ यूजिंग अ pressure cooker, that you are putting pressure so that the temperature at which water boils, that will increase. Now then this question, if you follow this basic idea, what they are saying is, in a pressure cooker, the temperature at which food is cooked depends mainly on which of the following. Okay. So pressure cooker में कितने temperature पे food boil होगा, food cook होगा, how will that be decided? The area of the hole in the lid. So lid here means the covering, cooker का ढक्कन, ठीक है? Temperature of the flame and weight of the lid. Do you think area of the hole in the lid that can decide? Pressure के बारे में सोचो, ठीक है? ये pressure cooker है, हमें बढ़ाना है pressure. हम वो ढक्कन सील करके बंद कर रहे हैं because we want to increase the pressure on the water which is there inside, जिसमें food है. So, which of these can have an impact on the pressure? Can the hole is from where the gases will escape. Can that hole have an impact on the pressure? Yes, right? Hole बड़ा होगा, gas ज़्यादा निकल जाएगी, छोटा होगा, कम निकलेगी, ठीक है? So pressure change हो जाएगा accordingly. The more gas which will be inside, the more pressure is going to be exerted. So this should be correct. Then temperature of the flame. Temperature of the flame again, it will heat, but pressure boiling से ही decide होगा, right? या ऊपर से जो exert हो रहा है. So this cannot really impact it because that flame is the same. That flame was the same for an opal vessel. The flame is the same for the प्रेशर कुकर. दोनों में सेम ही फ्लेम है, फ्लेम का टेम्परेचर भी सेम है, उससे कुछ चेंज नहीं हो रहा है.
जब हम लोग गैस को ज़्यादा करते हैं तो जल्दी ना कुकर.
दैट इज देयर, बट आप कंपेयर तो दोनों उसमें से करो ना. एक ओपन वेसल है, उसको आपने जितना टेम्परेचर दिया और एक प्रेशर कुकर है, उसको जितना टेम्परेचर दिया. अगर सिम पे बना रहे हो दोनों सिम पे बनाओ, देन यू कंपेयर, राइट. अगर फुल पे बना रहे हो तो दोनों फुल पे बनाओ, फिर कंपेयर करो. कैन दैट चेंज, राइट. जल्दी बन सकता है, राइट. इट्स नॉट अबाउट टाइम, दे आर आस्किंग यू अबाउट टेम्परेचर, राइट. आपने सिम पे बनाया, आप पानी चेक कर लेना टेम्परेचर क्या है. आपने फुल पे बनाया तो भी आप पानी चेक करना. इज दिस temperature of the flame, it can reach that temperature in a faster time, but it will not change the temperature. Water अगर 100 पे boil होगा तो 100 पे ही boil होगा, उसके बाद its temperature will not increase, because then that energy will be used for changing liquid to gas. ठीक है? Okay. Weight of the lid. So you know that lid on the cooker, right? So do you think that can make a difference in the pressure? Yes, of course. वो lid हटा के देखना कैसे कुक हो रहा है और उसके बिना कैसे हो रहा है. That is the whole point of that lid, is to put in more pressure. So whatever is changing the pressure, that will change the temperature at which the food is going to be cooked. So we don't want 2 in our answer. And if you can just identify this statement, this question will clearly give you an answer, which is C, 1 and 3 only. ठीक है? These things, even if you don't read them exactly, as you might not get to read them exactly, still if you understand the basic concepts, again which are there in your NCRTs, you should be able to solve these questions. अब ये एक बहुत बहुत कॉमन स्कूल लेवल का क्वेश्चन है, वाटर इज द यूनिवर्सल सॉल्वेंट, राइट. बट द प्रॉब्लम इज कि एग्जाम में समटाइम्स ये क्वेश्चन भी ऐसे लग जाता है. द मोर बेसिक अ क्वेश्चन इज, यू नो, द मोर डिस्कनेक्टेड वी आर टू दैट, राइट. बिकॉज वी आर रीडिंग एडवांस लेवल थिंग्स, we are reading adenovirus and its structure, we've forgotten about water, right. So that's why it's important to go back to the NCRT books to read such things. So this was a very simple question in that sense, and even if you've not revised it recently, तो थोड़ा question की sense से देखो, maybe you'll be able to solve it. Water can dissolve more substances than any other liquid. It is dipolar in nature. Dipolar or having a polarity means कि इसमें थोड़ा सा slight negative charge होता है, slight positive charge होता है, and that is basically because of its structure. The structure of water with hydrogen and oxygen is this. Oxygen is, you can say, a stronger molecule here, and oxygen kind of pulls the electrons towards itself. There are bonds which will be formed, and जो bonds हैं they are formed of different electrons. So ये oxygen खींच लेता है थोड़ा सा अपनी तरफ, यहाँ पे जो है negative charge हो जाएगा, hydrogen पे slightly positive charge हो जाता है. So it kind of has both ends. It has a positive end, it has a negative end. That is what it means by saying it is dipolar. Then it is a good conductor of heat. Do you think being a good conductor of heat has anything to do with dissolving things? Right? ये बिल्कुल unconnected concept है. Then again the other one, next one is also with heat. It has high value of specific heat. Unrelated. It is an oxide of oxygen, hydrogen. ठीक है? Again, this is factually correct, but what helps it dissolve more substances is, because of this polarity, it can attract different things. Negative है तो positive चीज़ों को attract कर लेगा. Positive वाला है negative चीज़ों को attract कर लेगा. जिसमें negative polarity होगी या जिसमें positive polarity होगी. So in that sense, it will be able to attract a wide range of things, and when they are attracted, they can stay in dissolved form in this liquid, making water the universal solvent. Okay. So this answer is going to be A. It is dipolar in nature. Going further, applications in everyday life. In this now, the questions which they ask, they are mainly going to have a current orientation. Current affairs में news items of, like we'll see examples of adulterations, of some new product, new application that's going to be asked. When one very prominent theme which has been asked in this particular section is contamination. contamination or anywhere where there is some product with some negative use, some banned product. Okay. And these kind of news articles also keep coming in the newspaper. The government has banned this, or there is a demand that this should be banned. So whenever there is any such point, any such news, you should note down the substance. You should note down कि क्या चीज़ है जिसको बैन करना है या कर रहे हैं या करना चाहिए. ठीक है. They usually don't ask you the negative impact or the problem or anything, but they do ask about things. कि ये है क्या? ये substance है तो what is it? We'll see that in the questions which are there. There are a number of questions here. This is the first one. Commonly used consumed materials and unwanted or controversial chemicals likely to be found in them. So we have lipstick, soft drinks, Chinese food. The question is asking which of the pairs are correctly matched. Okay. So lead in lipstick, soft drinks and brominated vegetable oils, and Chinese foods and monosodium glutamate or MSG. So this question has also come at a time where there was a lot of discussion on MSG. I'm not sure if you remember, but Nestle, Maggi, ठीक है, Maggi खाना बंद कर देना है, until you know Patanjali came and said हम अच्छी Maggi खिलाएंगे. So the idea, this was a very, very widely debated concept, that MSG, and it's there in all Chinese food and how it's bad and all of that. And there's reports that this MSG has a very specific stimulation for the nervous system. That's why it tastes good also, because a lot of nerves get excited, and ultimately it is also suggested that that might have some toxic impact also. So I think 3, at least at that point of time, if not today, would have been very easy for everyone to identify: Chinese food and monosodium glutamate. So in this, 3 should definitely be in your answer. And lipsticks and other such cosmetic products, they can have lead as an adulterant. So 1 is also correct. And so soft drinks do use brominated vegetable oils. This brominated or bromine concept is basically to maintain the citrus quality, or to lock the citrus flavor in soft drinks. So all these 3 are correct. Here again, approximation can work, but not entirely. Elimination or anything else doesn't work. So if you've read about something linked to it, or to maybe a related food product also, or related consumer good, then you can choose. Otherwise, you cannot really say. I think the second option was a little bit tricky or confusing, but first 2, I think identification was easier. Okay, this is now where UPSC, of course, wants you to read. This is a very, very common topic, but otherwise also your bottles which you buy, the products which you buy, or if there's any baby product or anything, they do mention this. ये बड़ी common चीज़ है, लिखा होता है BPA free. अभी देखना शुरू करना चीज़ों पे, plastic goods पे, they will write that this is BPA free. A bottle which you buy, that might write, या search करके देखना, BPA free products. Now, this is bisphenol A. It is a cause of concern, is a structural key component in the manufacture of which kinds of plastic. However, even when this was a common theme, the question is still very difficult, because the options are very similar. ये सभी plastics के ही नाम लिए हैं उन्होंने. Okay, they have low density polyethylene, polycarbonate, polyethylene, terephthalate, and polyvinyl chloride. So the options here make this a very, very difficult question. unless and until you've read a specific news on this, there is a lot of news coverage on this. And if you can recall, because ऐसे examples में confuse होना is also a little easier, right? कि polycarbonate था या कुछ और था, but the answer of what this is made up of is polycarbonate. So theme is common, but they expected you to go a little bit more into this. Again, a similar question of additive substance. So this is triclosan, considered harmful when exposed to high levels of for a long time, is most likely present. So ये basically है एक antiseptic, antimicrobial agent. So it is supposed to provide protection against growth of different microorganisms. Okay, so things which have this is used in many different products. Even they have clothes which have come, which they say कि these clothes are antibacterial or antimicrobial. ये clothes से में bacteria नहीं grow होगा. They are also using this product, but it's mainly in a lot of your toiletries. Toiletries like your toothpaste or your face cream, which again you want to increase its shelf life, that it should not get contaminated with some microorganism. So for that, this is added. Again, there are a lot of reports on this. Both this and BPA, they are considered to be having a very severe impact when they are long-term used on our endocrine system, the system of hormones. How hormones work, their response can be disturbed with such agents. This, because this is directly there, even with its good properties, it can have some negative impacts. And BPA, they say कि bottle में से, especially like there's one very basic concept कि plastic की bottle में गरम चीज़ नहीं डालते, right? You understand that, है ना? Right? उसमें idea, of course, is this: with that at those extreme temperatures, different forms of plastics, they can actually dissolve. So things or products which they are made up of, they can leach into that liquid or that water or whatever it is. So ideally that should not be done. Or this concept of, you know, microwaving in all different kinds of plastics. So plastics will be specifically mentioning that we are BPA free, we are microwave safe, because leaching can happen at higher temperatures. That's why long-term use of those products is discussed to be having a lot of problems. After this, we have probiotics. Again, not something which is in news, but otherwise very widely discussed. Having, in terms of dietary intake, increasing the use of probiotics in our diet, that's one point which is always mentioned. So let's see what is a probiotic. In our intestines, we have a lot of bacteria, and these bacteria are supposed to help us digest our food. Okay. If our gut, if our intestines are healthy, we'll have more of these bacteria. If it's not a very healthy system, there can be some disbalances and the bacteria can die. So we need to have these good bacteria in our body. They'll help us digest our food. So ये good bacteria मिलते हैं yogurt में. There can be some probiotic milk, cream, and there are many fermented foods. Fermented foods में you can have things like idli, and there are many drinks like kombucha which are fermented and which have these bacteria. Fermentation happens because of bacteria and yeast. So any food which is fermented, like dosa batter also, dhokla, all of these, they will have the good bacteria. So good bacteria is called probiotic. There are some foods which help in the growth of the good bacteria. These foods are called prebiotics. Prebiotics means, think of this कि bacteria को grow करने के लिए bacteria को कुछ specific food चाहिए. Bacteria need some special things which help them grow and flourish better, and those foods are called prebiotics. So we have garlic, banana, asparagus, wheat flour with the more fiber content, bran. So foods which help probiotics to grow, prebiotics. Foods which are containing good bacteria, they are probiotics. And this was a question on that. Again, a generic question in the sense of understanding, if you know the idea. Consider the following statements in respect to probiotics. Probiotics are made of both bacteria and yeast. See, when I talked about to you about fermentation, these are the two main organisms involved. So this statement is correct. Organisms in probiotics are found in food we ingest, but they do not occur they do not naturally occur in our gut. What do you think about this statement? False. We are just trying to help our body have or create something which it naturally has. There are a lot of bacteria living in our body happily, healthily, and helping us in digestion. So ye ho gaya incorrect. 2 shouldn't be there. Then probiotics help in the digestion of milk sugars. What do you think about this statement? Okay, I'll give you an example of this or an idea. There are a lot of people who cannot digest milk or who are not very comfortable with milk, right? But they can digest curd, right? Or you might have heard that curd is easier to digest. ये जिनको दूध digest नहीं होता, they can maybe try छाछ or lassi or something like that. Yes, you understand that, है ना? So why that is so is because that has probiotics, that has bacteria already doing the job of digestion for you. So these probiotics can help in digestion. Their job is to help in digestion. Milk is one of the basic substances which are going to be helpful, which are a little difficult to digest, especially as people grow older, they find it difficult to process milk. So in that case, if you have probiotics, that they will do the job. Okay? Okay. Again, reading newspapers and articles sometimes tells you about these things. Like this is again a clipping from the Hindu, where it's just this heading. So our probiotic beverage is good for health, right? You know about Yakult? Yakult milk, choti si bottle aati hai. That is a probiotic milk or milk with a lot of good bacteria, which people are drinking. The problem with that and on discussions like that is they put a lot of sugar in it. So people who were drinking it for good digestion, they actually increased their weight and their sugar levels because there was a lot of sugar in it. Okay? So hence this discussion. Again, what I'm emphasizing here is don't ignore a heading with a term if you don't know the term. You should know the term at least. That's how your preparation is supposed to build. Don't miss out on these things. Okay? Okay. Next is space. Yeah. Of this part also, basic science also he has taken. Ah, okay. Then that's good. You save me some time. Probably that's why he did not complete his part. Okay. Let's take a break here, right? Then we'll start with technology. Now coming to the technology part. In this, we have biotechnology. Actually, what we mentioned there in heredity and evolution, and this is all actually part of one topic only. There's no distinction here to categorize this. So the same concepts of how DNA is working, how RNA is working, that we are going to apply in different applications here. This is the first part, and this is somatic cell nuclear transfer. So it is talking about a somatic cell nuclear transfer. Somatic cell is any cell of the body other than the gametes. In our body, you have two types of cells. One is the gametes. Gametes are the reproductive cells, like your egg and sperm. And all other cells. Leaving these two, all the other cells, they are called somatic cells. Okay. So what is the application of somatic cell nuclear transfer technology? Without going into the technology, we'll go to the technology also, but let's just see if we can make sense of these options which are there. Let's try. Production of biolarvicide. You understand larva? What is larva? You know, butterfly, caterpillar, larva, right? The early developmental stage of these insects, that is the larva form. So biolarvicide means side, like insecticide to kill insects, pesticide to kill pests, larvicide to kill the larva. If larva is a problem in some case, to kill it, if you're using some biological agent. Bio means something from living organisms. So if you're using that, that will be a biolarvicide. So somatic cell and biolarvicide. Okay. Then manufacture of biodegradable plastics. Okay. Then reproductive cloning of animals and production of organisms free of diseases. Okay. Now in this, if I have to approximate, I feel that this larva and plastic, these might not have any link with the working of a somatic cell in any way. And producing disease-free organisms, reproductive cloning maybe, but let's see what this process is. This word cloning we'll talk about. When you say cloning, it means making an exact copy of an organism. Making an exact copy of something, like Xeroxing something, that is what is cloning. So if you want to make a copy of an organism, for that a somatic cell can be used. How that works is, see, normal reproductive process, there is an egg, there is a sperm, they combine, they fuse, we get a zygote. This zygote develops more cells, it forms the different parts of the body, different hands, limbs, organs, sab banke, finally you get a baby. And this baby has features of the mother who has given the egg and also features of the father who has given the sperm. The baby is not exactly like either of the parents. The baby is a combination of these parents. Okay. In terms of genetic material, how we look at it, we have said that DNA is transferred from one generation to the other. Right? When we were talking about genetic material, we said we are getting it from our parents. The form in which we get it, that form is chromosomes. Have you heard about this term, chromosomes? Okay. So chromosomes are basically made up of DNA. And DNA, you can say the whole DNA of a body, which is very large, that's actually divided into a number of chromosomes, and that's how it's found in our cells. So for reproduction, genetic material is transferred through chromosomes only. In human beings, this number of chromosomes is 46. For every species, this number is fixed. Okay. For different species, it will be different. Let's say sunflower will have a different number. Honeybee will have a different number, but एक species में fix है. Monkey की एक species में that same number is going to be there. Human beings में the number is 46. So during reproduction, how you get these 46 chromosomes is 23 come from the mother, 23 come from the father, and then when these two cells join, you get your 46 chromosomes. Okay. Why the properties of a person are a combination of both parents is because both are giving genetic material, both are giving chromosomes. In cloning, we don't want that. In cloning, we want properties of only one parent. That's the whole idea of cloning. So for this process, we use a somatic cell. Okay. Zygote has 46 chromosomes, and after zygote, all the cells of the body, your brain cells, your muscle cells, your blood cells, they will all have 46 chromosomes. So if I want to make a copy of one organism, I want ये सारा 46 एक ही parent से आए. It should not come from the two parents. So somatic cell I'll take, and somatic cell will have the complete set of chromosomes. They are present in the nucleus. For reproduction, for this zygote to divide and form the baby, you need an egg. Properties of an egg and the female reproductive system are important for reproduction. So we will need an egg also in this process. Okay, if this is a human cell, तो यहाँ पे 46 chromosome होंगे, egg में 23 होंगे. But egg can genetic material we don't need. We want only somatic cells genetic material. So what we do is we take this egg, we empty it, and इसमें हम डालेंगे somatic cell का nucleus with the 46 chromosome of the person who has given the somatic cell. This process is somatic cell nuclear transfer. This is given in class 12 NCERT. Okay, as such, cloning ka concept is something which is in news also. China had cloned a wolf, then this topic got highlighted. They had successfully done that. But otherwise, ये एक basic topic भी है, NCERT में covered है, animal cloning. Okay. So from somatic cell, you are transferring the nucleus to the egg. This egg can then be stimulated in the lab to finally form an embryo, और embryo से फिर बनेगा baby. This baby will be the exact copy of the organism who has given the somatic cell, because it has the same nucleus, same genetic material exactly. All features, face, hair, eyes, everything, because all the things are determined by genetic material, they will all be the same. Yes, all these keywords is important, but once you'll actually get to read it, ये automatically आ जाएगा, feed हो जाएगा. If you don't know this would have been, see, if it was asked in a particular year, it would have been somewhere in the current affairs news also, right? But if you've not come across something like that, right, then at this stage, okay, maybe going into a concept like that will not be an ideal thing. Because when it's just 4 weeks to the exam, the best thing you can do is just revise whatever you have already studied. Okay, so introducing new concepts, and especially concepts like these, which involve so many new terms, that's not an ideal strategy. You lose out on what you know. So revise what you know, and if you are preparing, then yes, you should know, okay, these are basic concepts. If they are in current affairs, they will need you to prepare them, and as such, basic idea, cloning is a very basic concept, NCERT level concept. Okay. In this case, again, approximation will take you somehow closer to the answer, but here the answer is reproductive cloning. When the word reproductive is used, it just means producing a new organism. That's the idea of reproduction. Cloning means making a copy, and that copy you can make through somatic cell nuclear transfer technology. Answer is going to be C. Of course, they don't ask you about the details of the procedure, but it gives you a little bit sense to help you understand that and remember that. Next is genome sequencing. Okay, so I told you this word genome. What is genome? What does it mean? We spoke about it a little while back. Complete genetic material, right? एक organism का complete genetic material जो है उसको बोल दिया genome. Now, if you look at the structure of DNA, DNA is made up of 3 things. If I write about DNA, this is deoxyribose nucleic acid. Okay, and it is made up of three things. There is a phosphate group, there is a pentose sugar, the name of the sugar is deoxyribose sugar, and there are nitrogenous bases present. This structure is fixed. Now DNA can come from bacteria, DNA can come from virus, from amoeba, from plants, from algae, from animals. Anywhere you have DNA, its composition is going to be fixed. This is going to be its composition. Within nitrogenous bases also, now further, if we look at it, we have purines and you have pyrimidines. Here you have adenine, guanine. These are the names of the two purines. See, you need to know this. This is the most basic idea on DNA. You don't need, again, like I told you earlier also, you don't need structures or formulas or anything, but you should know these things are there in DNA. Pyrimidines mein you have thymine and cytosine. We use standard abbreviations for these. Adenine, guanine. Then you have thymine and cytosine. Now, how this is arranged, this whole thing, like we saw double-stranded structure of DNA. I told you DNA is double-stranded. So, ये हैं दो DNA के strands. We are not making them in a twisted form. We are simply writing them in parallel structure. This has a sugar phosphate base, a sugar phosphate backbone. And you have nitrogenous bases. They can be in any random order. Okay, I've written it like this. It could be anything, C, G, any random pattern can be there. And what happens is purines join to pyrimidines. Adenines join to thymine. Guanine joins to cytosine. This is fixed. A के आगे T होगा, T के आगे A होगा, C with G, G with C. This sequence of nitrogenous bases is what defines our characters. Like these smaller parts of DNA, they can have different information. I told you DNA holds different information. Say this much part has the information for your hair color. The next part has the information of what kind of eye color you will have. We call these smaller segments or parts as genes. Okay, so this part is the gene for hair color. This next part is the gene for eye color. If we look at the complete DNA, or if we look at the complete genome of a person, we say that we can have like genes like this, we can have more than 20,000 genes. See, there are so many features. Like if you think about hair also, hair ka length, hair ka texture, hair ka color, hair ki graying, hair ki strength, so many features for each thing, so many different properties. So that is what makes this a very, very elaborate molecule, a very complex molecule also. So smaller parts, genes, have the responsibility of individual features, and we have more than 20,000 genes. If you look at the sequence of nitrogenous base pairs, जैसे ये है 1, 2, 3, if you start writing A, T, G, C, C, like that, this will give you information of different features. Like here TAC can tell you that this is for brown hair color. कि brown hair color चाहिए तो उसमें TAC is what is needed. If you need brown eye color, then GGC is needed, like that. This importance of sequence I am telling you. And the total nitrogenous bases which are present, if you talk about the whole DNA, it is in billions. So when you write the sequence of the whole genome, it's a very complex, it's a very challenging process, billions of base pair sequences are there, but when we do that exercise, we call that genome sequencing. Genome, complete genetic material, it's made up of a structure like this, and when we write that sequence, ki A will come, T will come, G will come, when we keep writing that, we say we are doing genome sequencing of that organism. So the question is on genome sequencing. There are a lot of genome sequencing projects which are always in news. The governments around the world are trying to do that, are trying to study DNA, are trying to see which gene has what sequence and what is a particular gene is doing. How is a gene causing a disease? They'll check for its sequence. So this question is based on that. With reference to agriculture in India, how can the technique of genome sequencing, often seen in the news, be used in the immediate future? First is, genome sequencing can be used to identify genetic markers for disease resistance and drought tolerance in various crops. Genome sequencing is for identifying sequences, okay? They've used the word genetic markers, you can understand this as genes, to identify genes for disease resistance and drought tolerance. Like maybe there is a plant in which a particular disease is not happening, the plant has some protection. So we can do a genome sequencing of its DNA to see what is that gene which is responsible for this resistance. What is, if there is a plant which is drought tolerant, we can check its genes to see कि इसमें ऐसा कौन सा genetic material है, कौन सा genetic sequence है, because of which it has its properties. Because ultimately all our properties are because of our genetic material only, right? So first point, do you agree this can be an application of genome sequencing? Yes? Okay. Next is, this technique helps in reducing time required to develop new varieties of crop plants. Can you think of how this can be linked? See, once you know a gene, right, we have technology through which we can add these genes to a particular plant variety. Suppose हमें किसी plant में disease resistance का property include करना है. What you can do is, you've done a genome sequencing of a plant variety which has disease resistance. You can take that gene and you can put it into another plant to create a new crop variety of crops. Okay, or you can, even if you don't want to add the gene, you can simply fertilize it also. You can do a hybridization of those two varieties because you know that this variety has a gene for drought tolerance. Okay, that will help you create new varieties, and because you already have the data available for genome sequencing, so you'll not need to find that out again. वो एक बार baseline data बन गया, उसके basis पे whatever properties you need, you can get that. So this is also correct. It can be used to decipher the host pathogen relationship in crops. Okay. Pathogen is the disease causing organism, host is the organism in which disease is caused. So how it can be used to identify the relationship is, like I'll give you the example of COVID. Okay, remember COVID-19 virus and how it had many variants, right? Different varieties of COVIDs were coming, alpha, beta, delta, omicron, right? So these viruses, the different variants, they showed different properties, right? Some were serious, some were less serious. Do you remember that? More deaths in one particular type of variant, less infection in the other. So how these viruses were different was that their genetic material changed. Okay, so if you do a genome sequencing of all these different varieties of viruses and then you check, then you correlate their genes with the feature. Okay, if this gene has changed, then it is more harmful. If the second gene changes, then it becomes less harmful. You can establish relationships like that, and it will give you a better idea that if a pathogen is acting in a particular way, why is it acting like that? So that can also be the logic of, or something which you can, the help you can get from genome sequencing. Remember this thing, sequencing means understanding or writing the details of the sequence. अब उस sequence की information के based पे you can have any kind of application, any kind of study which you want to do. Genome sequencing में we don't make anything. We don't make products, we don't make new varieties, we don't do anything. We only have this data for different sequences, and based on that sequences, then other applications could be there. This again is a very, very commonly used terms. We have 2-3 projects running in India also to do human genome sequencing, where they want to understand what are the genes in different human beings in India. Okay, there are projects like there's this project called IndiGen. So they have a target that some 10,000 people, their genetic material, they are going to evaluate in this way. So ये common recurring news pattern है. This is a 2017 question, but you can very well have questions again. If you understand the concept, see, you will never read all these specific applications. ये आपको खुद ही apply करना है. The only thing is, if you understand the theme, you will be able to apply this, right? This portion of biotechnology will need one basic line of preparation at your end before these questions you can solve. And that you will need to do because, again, very common in your current affairs. Yes, sir.
Species of plants, for example, one plant from desert area and one plant from tropical area.
The question is asked is, can we do genome sequencing in two different varieties of plants? Sequencing simply means you write the sequence. So if you have 2 plants, you take one plant's DNA, you start writing, that is this plant's data, desert plant. The other plant, says tropical plant, you take its DNA, you start writing. We can do sequencing for all species which we have, right? There is no limitation to that. So आप कितनी भी species का कर रहे हो, of course, you can, you'll have to do them separately. And there is a possibility that there can be some genes which are common also. Once you have the data, you can establish relationships also, right? That if there is a linkage, if there is some part of the sequence which is common, you might understand that they have evolved from common ancestors. जैसे आप human DNA देखोगे और ये डेनिसोवन का DNA देखोगे, ठीक है, जो मिला है, right? उसका tooth मिला है and all of that. There is some commonality and there is some variation. Commonality indicates that yes, we have evolved from them and now we've become different because the genetic material has changed, right? आप monkeys का देखोगे, monkeys से भी match करेगा कुछ. That also indicates evolution. Okay. Next is this question on GM mustard. This question is a factual question. Sometimes current affairs में से direct current affairs fact can also be asked. Though concepts will take predominance, but again fact भी पूछ सकते हैं, like in this rare question. Now GM mustard की story ये है. GM mustard has been around. GM means genetically modified. When we make any genetic change in any particular organism, that becomes genetically modified. Now this was created many, many years ago, and the committee which was set up to approve these crops, that cleared it also. So everything was ready. The government also thought that okay, now we are going to approve it, it is going to be grown. But then there was some objection. The court went to the case went to the court, where they said that this is not very good for our environment, so you should not be growing this crop. And then this was stopped. As of today, we are not growing it. It's ready, but it is under regulation process. So while it was in court, and that has been for many, many years, but whenever the case comes around, this is then again becoming a part of very, very major editorials, continuously large number of articles written on this. So this is one of the crops which is closest to cultivation in India. As of today, we only grow one genetically modified crop. India mein, there are a lot of crops which we have created, but GM crops mein ek hi crop hai, which is BT cotton, which we grow commercially right now. Rest everything is in regulation. सबसे ज़्यादा hit crop है उसमें से GM crop, GM mustard. ठीक है? So let's read about the question. With reference to genetically modified mustard developed in India, consider the following statements. GM mustard has the genes of a soil bacterium that gives the plant the property of pest resistance to a wide variety of pests. GM mustard has genes that allow the plant's cross-pollination and hybridization. GM mustard has been developed jointly by IARI and Punjab Agriculture University. So one very commonly known fact about GM mustard is that it has been prepared in the Delhi University. So if you're reading about this when it was a current affairs theme, then this would have been one point which was highlighted. So this statement is incorrect. And this is a very simple statement for this during that time when it was being discussed. This is also a defining statement. If you look at the answer, sirf isse hi answer aa jayega. Can you see your options? All the options have 3 other than this. The first statement here, GM mustard has genes of soil bacterium which gives the plant pest resistance. This is true for BT cotton. BT cotton is a variety of cotton which has a gene. Ye BT is for the bacteria, the name of the bacteria. And this bacteria has provided cotton with the special property of getting protected from pests. Ek pest hai. We have a question on BT cotton also. Bollworm. ये पेस्ट का नाम है, वर्म का नाम है, which attacks cotton, and एक bacteria है which produces certain toxins which kill this bollworm. So what we did was we took that gene from the bacteria and we put it into the cotton crop. So cotton developed pest resistance. But this is not the case of mustard. Mustard, this GM mustard which we produce, that is a high yield variety. That is a high productivity variety. And how it is showing high productivity is that because it allows cross-pollination and hybridization easily. So you can hybridize it, you can fertilize it with other crops with better properties. That is the special gene which you've introduced in it. So only second option here is correct. The first one fits for BT cotton, and this last one is a pure factual statement which is again incorrect. Answer is B. Okay. I want to tell you a little bit about the basic concept of preparing these technologies. Whenever you have any technology development taking place, you need to first know the process, the basic idea, process, or the basic principle on which it works. Then the other significant development would be example of use. Like suppose there is a technology and there's a product being made on that. So that example of the product, that will be important. And in this, if there is any development which is taking place in India, that becomes even more important. The chances of a question being asked to you based on a particular technology is, the likelihood is if it is an indigenous technology, the chances of that being asked is going to be very, very high. So that you will have to specifically track. Ki, okay, genetically modified crops. Are there any crops being formed in India? Those crops you should know about. You should know their names. Prelims mein this much usually works. For mains, you will also need to add your angle of advantages and disadvantages. So this will kind of complete your topic. How is it supposed to be used, and if there are any problems with it, that you should also know. Okay. Now, after this, what we go to is the next question, which is now a little bit of a question again, which you can say general understanding or concepts of how DNA work. And this question is also a question which is not on one theme exactly. It's not on one topic. It's picking many different ideas. So just read this question, because we've had a lot of discussion on DNA. See if you can think of anything making sense to you. With reference to recent developments in science, which of the following statements is not correct? Okay. First is, we are looking for an incorrect statement. Okay. So let's see. Functional chromosomes can be created by joining segments of DNA taken from the cells of different species. Pieces of artificial functional DNA can be created in the lab. Piece of DNA taken out from an animal can be made to replicate outside a living cell in a laboratory. Cells taken from plants and animals can be made to undergo cell division in laboratory petri dishes. So this question is slightly confusing, similar kind of statements. Let's take one statement at the time. Okay, you think the answer is D, so let's start with D. Cells taken from plants and animals can be made to undergo cell division in the lab. Okay. We have a technology called tissue culture. For plants, this is something which is very commonly used. What we do is, we identify the growing parts of the plant, growing tips of plants. Usually the ends of the leaves and the stems, they are the growing parts, where the cells are still capable of growing. So we take cells from there, and in the lab, we provide these cells with all the proper nutrients, the hormones which are needed by plants to grow, everything, ideal temperature, ideal pressure. And what we can experience is that under ideal conditions, proper conditions, these cells will start growing into small plantlets. They can be then taken and grown in the soil, grown in the pot, and they can form full plants. So this is very commonly done where fruits and flowers in horticulture, basically tissue culture is used. We can grow plant cells, we can grow animal cells in the lab. Growing means one cell forming more cells, and that is going to happen through cell division. So that is very much possible. So that means your option D can happen. See, cells taken out from the plants and animals can be made to undergo cell division in the laboratory petri dishes. So this we can do. Then a piece of DNA taken from an animal can be made to replicate outside a living cell in a laboratory. This is also correct. We can take DNA, we can make copies of that DNA. We can even prepare artificial DNA. Okay, DNA, because we know what DNA is made up of. Okay, we know that there is a phosphate group you need pentose sugar, you need nitrogenous bases. So you bring all of that, you can prepare an artificial DNA. If you already have DNA, you can make copy, process which we mentioned. So DNA replication, that can take place in the lab. So we have D also is possible, C also is possible, B also is possible. The only thing which we cannot do, as of today at least, maybe at some point we have technology for this, that is creating a full functional chromosomes. Okay, we can have DNA, we can add DNA from different species. Like I told you, genetic modification, genetically modified crops. Bacteria का gene लिया, cotton में डाल दिया. That we can do. But we cannot create a full functional chromosome. Just one part we can remove or add. That is called editing. Editing we can do, preparing a full chromosome, the whole chromosome in its entirety in the lab, that we cannot do. As of today, we have no technology to do that, so this is not correct. But in attempting a question like this, approximation and other options had to come primarily. Okay, so each of these three statements you had to identify and understand that they are correct. That's the only possibility of reaching to this. And again, none of these statements is specific on any topic, just on a generic understanding of DNA and technologies around it. Okay? So A is going to be the answer. If you're able to answer a question like this, then you are going to be at a very good place when it comes to the total number of questions you attempt in the exam. Okay, this is RNA interference technology, which has gained popularity in the last few years. Okay, so let's see, we did this process. DNA से RNA बनेगा, RNA से protein बनेगा. The term used for this is gene expression. Because this DNA is actually a segment of DNA, not the whole DNA, and just one gene will undergo this process of gene expression at one time. For a gene to work, protein formation is needed. Like we said, that hair color, there is one part of DNA which has the information for hair color. For that hair color to actually be formed, protein formation is needed. So, if we can stop the formation of this protein, through any method, if we stop this protein being formed, we will stop the gene from expression. Then the gene will have no impact. Because gene में सिर्फ information है. DNA is just information. The information has its impact when a protein is going to be formed. So, we use the terms to describe this. This is transcription. RNA to protein is translation. If we either prevent this from being formed, or if this is formed, then we prevent proteins from being formed, we'll stop the gene from working. This process is called gene silencing. If you don't let protein being formed, you can say you have silenced the gene. Okay. Now your options are either DNA to RNA you stop or RNA to protein you stop. Either of the two you have to do. There is a very successful technology here which has also won a Nobel Prize, which is also given in your NCERT, which is called IRNA or RNA interference technology. And this RNA, like I told you earlier, it is a double-stranded RNA. This is an exception of RNA structures. This IRNA is naturally present in our body whenever our body doesn't want a gene to work. Suppose there is a defective gene which can cause cancer. Your body will try कि ये gene ना काम करे, then it's better, right? Otherwise the person will have cancer. So we have inbuilt mechanisms in many cases which work very well. But now we have used this to make a technology. If we want to stop any feature, we can use this IRNA or RNA interference. What it does is, when DNA forms RNA, mRNA in this case, this IRNA attacks the mRNA and prevents it from working. Once this mRNA is attacked, it will not form any protein. This process comes to an end here. No protein means no impact of the gene. So if you have any disease which is because of defect of a particular gene, like suppose there's a cancer because of a defective gene, you can stop that. If there is any other property which you don't want to express, which you want to change, you can silence any gene which you want. Now this is a question again which is asking about applications of the process. Okay, so this is there, this idea is there in the class 12th NCERT. I'll read the question for you. RNA interference technology has gained popularity in the last few years. Why? It is used in developing gene silencing therapies. Okay, this is a direct statement. So one is going to be there. It is used in therapies for treatment of cancer. Okay, we can have cancer because of defective genes. So yes, that will also be there, giving you the answer. 1, 2, and 4. Okay. The third statement is it can be used to develop hormone replacement therapies. See, hormone replacement means that in some person there is a deficiency of a particular hormone. Like somebody has diabetes, the person has no insulin in his body or very less insulin, so the person takes insulin. I'm sure you know there are a lot of people who take insulin, right? Heard about that. That is hormone replacement. Okay. So this does not have anything to do with hormone replacement. This is influencing how genes work, or it is stopping genes from working. So replacement it will not work. The last option is it can be used to produce crop plants that are resistant to viral pathogens. Okay, maybe there is some property of a crop which is created because of a gene, and that property is the reason why the pathogen is coming to attack. Okay? Maybe the color, maybe the sweetness, something like that. If we stop that gene itself, whatever feature is attracting the pathogen, if that gene is stopped, then those pests will not be able to attack. Silencing that gene will make them resistant to the pest. So this can also be one application. Okay. So A, 1, 2, and 4. Next is gene editing. See, out of the total genome of a person, okay, if you can add, delete, or replace any gene, that process will be called gene editing. Okay, like that example of taking bacteria's gene and putting it in cotton. That is editing the genome of cotton. You have changed the genome of cotton, you have edited the genome of cotton. Now this gene editing or words like genome editing, they will again be in very continuous news. And the technology or the tool which is used, the most successful one is Cas9 protein. There is a gene editing tool, for editing DNA. You have CRISPR. Again, very important because Nobel Prize winning technology. Well, this question is actually from before it won the Nobel Prize. And this was again in recurrent news. Even now, this year also, CRISPR is very important because the first application of CRISPR on a patient with sickle cell anemia, that has come now. What they do is, suppose somebody has a disease, like the example of sickle cell anemia as it is being used for this. Sickle cell anemia is a disease because of a defective gene. There is one gene or one part of DNA which is defective, and that can be edited out, that can be removed. For gene editing, or if you talk about CRISPR technology, gene editing there are many ways in which we can do that. This is the most simplest and most successful technology. Again, winning a Nobel Prize kind of further increases its stature. So isme there are two components. There is a DNA cutting enzyme. There is one part of CRISPR which can cut DNA. That is your Cas9 protein. And there is a guidance molecule. Why you need a guide molecule is, like we've discussed, DNA is very big. DNA is very large. That large DNA, out of that, just one specific gene you have to cut. आपको पूरे में से ढूंढ के ये सिकल सेल एनीमिया वाला जीन निकालना है. So you need a molecule to guide and reach that specific location. That is a special RNA molecule. So whatever gene you need to identify, for that you can prepare this. This is called, full form is single guided RNA, and this is also an exception as this is also a double stranded RNA. So CRISPR is a gene editing tool. It is also called molecular scissors. It is having two parts. One is a guidance molecule. This guidance molecule will take the system to that particular gene and the cutting enzyme will cut it. Once it is cut open, फिर आप उसमें add कर सकते हो, आप उसको deleted छोड़ सकते हो, you can replace it with a different normal functional gene. All options are open once that cutting has taken place. So this very important, very commonly in news, current affairs. So this was a very easier question in that sense because of the sheer prominence this Cas9 protein had in the news. What is Cas9 protein often seen in the news? It is a molecular scissors used in targeted gene editing. In this question again, no scope of any kind of approximation or guesswork or anything like that. Biosensors used in accurate detection of pathogens, genes that make plants resistant, herbicidal substance, synthesized in genetically modified crops. You cannot predict. They are all in line with, all these things can happen with proteins, different kinds of proteins. So either you know it or you don't know this question. Yeah. See, genes or DNA is present in all the cells of our body. Okay. So it will not make a difference where it is being taken from. The cells of your eyes, of your blood, of your hair, of your liver, they all have same DNA. Because ultimately they were all made up of, they started with one cell, zygote. So when you are collecting DNA segments for say genome sequencing, you will choose any location where it is easier to extract. And the most commonly used sample is the blood sample. Blood cells can be used. You can also use hair cells or cells from the inner wall of your mouth, the skin cells of the buccal cavity, the cheek. So they are also easy to remove, but the most durable and where you get sufficient amount of cells, that is blood. Okay, another question on gene editing. Now, if you think of gene editing and bringing a change in a particular organism's genome, you have to think of different stages also. Like, we spoke about the two cells, somatic cells and gametes. Okay, now think of gene editing in each of these aspects. If you do editing in a somatic cell, like if I have sickle cell anemia, there is something wrong with my red blood cells, and you change my red blood cells, or you edit my red blood cells, that will be a somatic cell change. Okay, but if there is obviously a chance that my sickle cell anemia can pass on to my children, because genetic material is inherited from one generation to the next. So in that case, if you just change my red blood cells, my children are not protected. For that, my gametes will have to be changed. If you change the eggs and sperms of people, then when fertilization takes place, those changes will reflect in the children. So gene editing may, when you're changing genetic material, you can change both somatic cells and gametes. But if you want it to reflect in the next generation, the change has to come in the gametes. Or, you know, when cells are forming gametes, at that stage, if you make a change, then it will show in the next generation. Another possibility is that when the zygote is formed, it is dividing into embryo, but this embryo is now only a few cells. At that stage, when all the organs and all the parts are not fully developed, at that stage also you can introduce a change. Because then automatically that change will go into the, like suppose in the embryo cells are still being formed, blood nahi bana hai. That stage pe you change the cells, so blood automatically will be formed properly. The correct cells, the proper red blood cells will be formed. So embryo ke stage pe, when it is supposed to be done, it will have very good results. It can also be done at the stage of gametes. And if you do it in an adult, then you might need to do it regularly, right? Because the cells will die, then you will have to give that change again, because cells might start forming from the original pattern, the defective pattern. So this, something like this question is talking about. And it is also talking about stem cells. So let's read the options, then I'll tell you about stem cells also. Consider the following statements. Genetic changes can be introduced in the cells that produce eggs and sperms of prospective parents. Does this sound okay? We are looking for the correct statement here. Yes? No? Okay. Yes. We can make genetic changes, we can edit, we can modify the cells which are supposed to produce eggs and sperms. This is correct. And a person's genome can be edited before birth at the early embryonic stage. Yes. This is in fact ideal, this is desirable. This will make sure that there is a lifelong change, or that genes impact will always be there. Because you have modified it at a stage where cells are not completely formed. They're still dividing, they're still forming, organs are still being formed. So this is desirable. And now, if you identify that both these statements are correct, there's only one option you can choose. The problem with this question is the third statement. Though UPSC by design does not want you to know the third statement, but that third statement was very disturbing for a lot of people. And those people who understand these two statements, they were also not able to attempt this question because they did not understand this. Human-induced pluripotent stem cells can be injected into the embryo of the pig. This is 2019. At that point of time, this was not in news. 2024, this is a very super hit news, biggest achievement of biotechnology. Okay, yes.
At the gamete stage, if we will do gene editing,
Yes.
then hello, yeah.
G.
Then after the means next generation is safe, but
Yeah.
after that generation,
See,
the coming generation, how?
if you change the gametes, okay, then that generation is safe. You have removed the gene, right? But the generations after that, they will also get that new genetic material only. Because defective gene आपने निकाल ही दिया, it no longer exists. So there is no chance of that disease coming in any subsequent generation, because now that defect is not there, right? And genetic material transmission is in continuation only, so it will not work like that. The last statement here, human-induced pluripotent stem cells. So stem cells are cells of the body which can divide, that means form new cells, and change form. Like think of the zygote. Zygote was one cell, Today you have a billion cells in your body. These billion cells have been formed by the zygote. Zygote divided into 2 cells, 4 cells, 10 cells, and then these cells changed form. Koi stomach ban gaya, koi lungs ban gaya, liver ban gaya. All these organs were formed from there. Okay. So any cell which has this property, even when you are born after that, like in adults also, there will be some stem cells. Like you have an injury, you lose blood. So your body is supposed to prepare new blood to compensate for that, right? And when you are injured, your skin will also disintegrate, so new skin will be needed. As such, you might have observed that the skin keeps shedding, right? Once a month, the skin is shedding and you will have new skin cells. The liver is working a lot, liver is disintegrating, your body will need to repair that, create new liver cells. So we have stem cells in our body which are like reserve cells that when something is needed, they can change into that type. Being pluripotent means a cell which can change to all types of cell. Human induced pluripotent stem cells means these are cells which we can create in the lab, which will be stem cells. That means they can divide, they can form more cells. Like from one cells, maybe 1000 cells we can form, and we can have these cells change into all types of cells. You can take these pluripotent stem cells, you can make muscles also from that, you can make blood cells also from that, you can make heart also from that. So what was done in this experiment, that they took human stem cells, they put it into the pig, then that pig developed with human cells, and the pig developed a heart. That heart was extracted from the pig and given to a human being. Okay, now they've done it with the kidney also, but the big significance is that the person had heart failures. Okay, so he was supposed to die anyways if he did not get that transplant. He got that transplant and he lived for 2 months with the heart of the pig. So this opens so many avenues for future transplant options. Because we have limitations of living donors. There are a lot of people who have some organ failure, but there is no donor which is matching with them. So if we can use animals in this, like pigs are very close in terms of how their cell structure is arranged to human beings. So if we can use organs from pigs, we'll be able to save so many lives. Okay, so if you can take an organ from a pig, some cells can definitely move from one place to the other. Okay. The important point for this question and attempting such questions is options are there for a reason. And if options are helping you, you have to not get demoralized with the question without looking at the option. A lot of people do that. Ki, yaar, I don't know this. Ye pig kahan se aa gaya? Right? So let's leave the question. Don't do that. How your brain works on that final day of the exam, how careful you are about reading each and every statement, that is really going to change your result in the exam. How you study the whole year round, that is definitely important, but the final day is equally important. And MCQs will require a lot of presence of mind. This is a very different game, so you have to be very careful. You read the first statement, you know it is correct, please go and eliminate right at that moment. You have to do these multiple choice questions in steps. This is very important. आपको लगा first तो सही है, 2 options निकाल दो उसी time पे. So that you get that confidence that, okay, now I am 50% close to the answer. Okay, that's how you do it. And even if you don't know something, you might still get the answer. Okay, so here the answer is D, 1, 2, and 3. Then next is pro-nuclear transfer. This is again a reproductive technology, somewhat similar to how you did a somatic cell nuclear transfer, but objective is different. In the context of recent advances in human reproductive technology, pro-nuclear transfer is used for. And mitochondrial therapy, same concept, 2 questions being asked in 2 years. So look at this image. We have been talking about DNA present in the nucleus. Okay, we've been talking about chromosomes. But other than chromosomes, there is another piece of DNA in mitochondria. Do you know what is mitochondria? Powerhouse. Yes, everybody knows what is mitochondria. Okay, it is a structure present in the cell and इसके पास भी एक अपना छोटा सा DNA होता है called mitochondrial DNA or mtDNA. So if this DNA is defective, then there is a possibility that there can be some disease. And the unique point of this is inherited only from mother. Like the chromosomes we have studied are being contributed by both the parents. 23 from egg, 23 from sperm. Mitochondrial DNA is not like that, it's coming only from mother. That means if a mother has this defect, it's definitely going into the next generation, because all the children are getting all their mitochondria from mother. So we are using a technology where we will change the mitochondria. How we can do that? Okay, this is the egg from the mother who has defective mitochondrial DNA. Now we are putting a sperm into it. Basically we are fertilizing it so that a zygote is formed. This is another egg which you are getting from a donor, a female who is normal, who does not have mitochondrial defect, and here also the sperm is going to go so that you have two zygotes. Now this zygote, because all the mitochondria in this zygote have come from the donor, so ye hoga normal. Same case here, this zygote will have abnormal mitochondria. Because mitochondria come only from mother, even if the sperm has been injected, it will not impact the mitochondria. Then what you do is, you do that same process of removal of nucleus. You remove the nucleus from here. This nucleus will have the 46 chromosomes. Okay, the nucleus has 46 chromosomes, 23 from the egg, 23 from the sperm, and you put it into the other egg. एक जगह से nucleus ले लिया, एक जगह से egg ले लिया. The mitochondria are present in the cytoplasm. There are two main parts inside the cell. When you will study cell structure, you will understand that. एक होता है nucleus, जिसमें chromosomes हैं, and the rest of it has all the other structures called cytoplasm. Mitochondria is present in the cytoplasm. So from the donor, you took this cell without the nucleus so that the healthy mitochondria come, and you took the nucleus with the 46 chromosome from the parents, the two people who have given egg and sperm, and the transfer will work. Now in this, there are 2 points or 2 terms which you need to know. This has also been in news very regularly. This was first done in UK, then it was done in China. There is a lot of discussion, and there is a lot of research going on mitochondrial diseases also. That what mitochondrial DNA, what kind of diseases it can cause. And there are impacts on this defect on common diseases also, like diabetes. They are trying to draw some correlation कि mitochondrial defect से हो सकती है. The case which we did, where you used a zygote, okay, to change mitochondria, or mitochondrial replacement happened at the level of a zygote. This is called pronuclear transfer. The zygote's nucleus, okay, because that's a very early stage of life, it is called pronuclei. So when you are transferring zygote's nucleus to another zygote, it is pronuclear transfer. And there is a possibility that you do it at the stage of the egg also. Okay, you can do it at the stage of the egg. Replacement pehle karoge, sperm can come later on also. Process will be the same, But here the term which will be used is maternal spindle transfer. The egg's nucleus is called maternal spindle. Okay, so if you use a fertilized egg or a zygote, it is pronuclear transfer. If you use the egg and once the transfer has happened, then you can bring in a sperm for fertilization, it's going to be maternal spindle transfer. No, this will be found in all animals, all higher advanced animals. They will all have this process, or they will all have mitochondria. And of course, ye treatment of mitochondrial diseases, that makes sense that we do it only for human beings because it's a complex process. But problems and working of different structures, that will be in each case. Ab question dekh lete hain. See, question mein again, we have gone into the technique, we have gone into how it works, but they are not asking you that much detail. Okay. They usually just ask that what is this term associated with. Okay. So let's see here. In the context of recent advances in human reproductive technology, pronuclear transfer is used for fertilization of egg in vitro by a sperm donor. So in vitro, yeah, jisko aap bolte ho IVF, you would have heard about IVF. This is when you do this process of the egg and sperm joining in the lab. Okay, so this is not what pronuclear technology is about. Genetic modification of sperm producing cells. Again, we are not changing anything about the sperms here. Then development of stem cells into functional embryos and prevention of mitochondrial diseases. Okay, so you should know that this term is associated with mitochondria, and if you know that, you'll get to this answer. The prevention of mitochondrial diseases. Okay, this was a question which they asked in 2020 and again in 2021, next year. In the context of hereditary diseases, consider the following. Passing on mitochondrial disease from parent to child can be prevented by mitochondrial replacement therapy either before or after in vitro fertilization of egg. Right? Before भी कर सकते हैं, fertilization के बाद भी कर सकते हैं. The child inherits mitochondrial diseases entirely from mother and not from father. This is the special point of mitochondrial DNA. Both these statements are correct. Answer is going to be C, both 1 and 2. Okay. This is on BT cotton now. So I told you BT cotton has pest resistance, and the resistance against which pest? It's bollworm. So this is the name of the technology, BallGuard. This is how the technology is sold. So BallGuard 1, BallGuard 2 technologies mentioned in the context of developing genetically modified crops, clonal propagation of crops. Clonal or word cloning means copy, no change, right? So here you are changing, you are adding bacterial genes. Production of plant growth substances, production of biofertilizers. No. So whenever there is an article discussing GM crops, should they be used, should they not be used, there the mention of the technology is there. Answer is going to be B. Then you have recombinant vector vaccines. With reference to recent developments regarding recombinant vector vaccines, consider the following statements. See, vaccines were a very big buzzword during the COVID-19 period. Okay, pandemic, vaccine, which vaccine work, which doesn't work, how vaccines are formed. So a lot of questions during those time periods have been asked by UPSC also. They've asked a number of questions in prelims, they've asked questions in mains also. Again, themes mostly they're going to be picking up from current affairs only. Okay, that's how it will work. So I'll give you a very basic idea of how vaccines work, and then we'll see what is this recombinant vaccine. For a vaccine to work, we'll use the concept of B cells. We studied how B cells are attacking the pathogens which are coming into the body. When B cells attack a particular pathogen, or the word antigen was used, when they are attacking it, they also develop memory for it. Okay. They remember that antigen, and they are going to remember it for any future attacks. Okay, so एक बार कोई चीज आ गई body में, once a pathogen is there, the body's immune system, it remembers, and the next time it provides a better protection, a more severe protection. So we can use different methods to make vaccines based on this phenomena. For the objective of a vaccine is to create this memory in the body, जिसको हम simple words में बोलते हैं, you have a immunity for a particular disease, right? Like if you've had chicken pox, you might have heard that most people don't have chicken pox again in their lifetime. And there the term which can be used is, I have immunity against chicken pox. And this is what we try to create through vaccines also. We are trying to create this memory. For this, we have to send in the pathogen. But we cannot send in the pathogen in its real form. जैसे अगर if you want to make a vaccine for a virus, you cannot send the virus as such. Then the person will get the disease. What is the point of that vaccine if you give the disease itself? So we change the form. We give this antigen, but we give it in a different form, so that memory is there. B cells will still do their job. They will remember some part of it, but there will be no disease. Okay. And one such possibility is, you take antigen from one pathogen, or not the antigen, the gene producing antigen. We have studied that everything, all features all our properties are because of our genetic material. Information for that is in the genetic material. So we take the gene producing the antigen from the pathogen. जैसे COVID-19 का example देती हूँ. COVID-19 वायरस का वैक्सीन बनाना है हमें. Okay? We take this COVID-19 virus, and from that we find which gene is producing the antigen. We take that gene and we put it in another virus. In this case, we call it the vector. Vector means carrier. This is going to be the carrier of this gene. This is the gene which we want. So we put it in another vector, and this vectors, this will also have genetic materials. Say if we talking about DNA, तो इसमें भी DNA, gene is also DNA, they will combine. Why they will combine is because DNA structure is same everywhere. We spoke about structure of DNA, phosphate group, pentose sugar, nitrogenous bases, they are the same. So plants में भी वही DNA, animal में भी वही DNA, bacteria में भी वही DNA, fungus में भी वही DNA. Everywhere the DNA is the same. So you can bring DNA from two sources and it can join, right? Like we've been talking about how we are putting bacteria's DNA into cotton. How it is combining, how we have joined that is because chemically it is the same. Using the same concept, we can get the gene from one pathogen against whom we are trying to make a vaccine, and we can put it in another virus which does not cause the disease. The logic of using this other virus or this vector is, ये कोई disease cause नहीं करता. So the person will not have the disease. At the same time, this gene will go in the body, it will produce the antigen, it will stimulate the immune system, and they will be protecting the body against that diseases in any future attacks. This is the idea of a recombinant vector vaccine. You have to read this very thoroughly. You need to know how vaccines work. This you will find in class 11th NCERT biology book. It's given in a lot of detail very nicely. So you can use that and read the different types of vaccines also. Types of vaccines are important. So this they have asked one type, which is the recombinant vector vaccine. Why we are using the word recombinant is because DNA comes from two sources. ठीक है, एक तो जो डिजीज वाला पैथोजन था उसका जीन लिया और एक दूसरा हार्मलेस वायरस था. So genetic engineering is applied in development of these vaccines. Genetic engineering simply means that when you can alter any genetic material. When you alter any genetic material, you change any genetic material, that is genetic engineering. So this is a very generic term which is correct. You're making something which is a recombination, so you use a genetic engineering process, one is correct. And bacteria and viruses are used as vectors. This is also correct. This bacteria and virus, why they are used ideally, we can use any organism. Recombination तो किसी भी जगह हो जाएगा. But they are very easy to grow in the lab. Okay, they are very easy to deliver as a vaccine. They will reproduce very quickly. So we can have a large number of these bacteria and viruses. So they are ideal vectors when we are delivering vaccine. Okay. You know about Covishield? Yes. It's in news these days again. So Covishield is a recombinant vector vaccine. इसमें एक पार्ट आया है COVID-19 वायरस से, and the second vector virus which it is using, that is adenovirus. And then it is delivered as a vaccine. Viral vector vaccine. This question, because this was a very prominent news, if you understand the basic concept, just by a very superficial reading also, this question was very easily solvable. Answer is going to be C. Then ACE receptor in the context. Again, 2021 COVID time related to COVID. COVID में, this is the virus, okay, COVID-19 virus. I'm sure you remember this kind of structure of COVID, the crown-like structure. So these are the proteins of the virus, the spike proteins. How this virus would attack is, this would go and get attached to a very specific location on the cell. The cells of our body, they had ACE2 receptors. Okay, these are just names of structures which are present on the cells, and this spike protein fitted very well in this receptor. So what it did was, spike protein जाके receptor में अटक जाएगी, receptor is not going to work. It blocked the function and that's how it killed the cell. These ACE2 receptors, they are very important for normal cell function. But when the virus attached to it, they were not able to function normally, and that's how this virus damaged the cell. What they have asked you simply is that how is this term related? Genes introduced in genetically modified plants, this is not a gene. Development of India's own satellite navigation system, no. Radio collars for wildlife tracking, spread of viral diseases. Again, either you know this question or you don't. No approximation or anything would work here. But during the time of COVID, this was very, very common information. Maybe not today, but at that time it was. Okay, then another question on vaccines. Yeah. It is definitely because of the crown-like structure. जो corona word है, that means crown. And the crown is the spike protein which is there. In a lot of virus structures, the protein is the proteins. All viruses will have their proteins. But many a times this protein code, this is inside the structure of the virus. Corona में वो बाहर था. That's why it's named corona. And this kind of becomes suitable for that attachment. Yeah. Okay. So in the context of vaccines manufactured to prevent COVID-19 pandemic, consider the following. Serum Institute of India produced COVID-19 vaccine named Covishield using mRNA platform. So if you're following the news of Covishield, can you tell me if this statement is correct? Okay, one thing is कि अभी मैंने बताया कि कोविशील्ड कौन से टाइप की वैक्सीन है, राइट? 2 मिनट हुए, तो it is not an mRNA vaccine. Okay, it is a vector vaccine, right? So ये statement है गलत. Answer होगा B, 2 and 3. ठीक है? Okay. Sputnik 5 vaccine is manufactured. Sputnik 5 and Covishield same technology, vector based vaccines. Then Covaxin is an inactivated pathogen based vaccine. Yes, Covaxin is an indigenous vaccine made in India. Inactivated में क्या होता है? You take a virus and you kill it. Okay, think of it like a dead body, and then you send that dead body in the vaccine. It has been observed that B cells can even identify that dead pathogen, that killed pathogen. Okay, because some parts, say it's cell membrane, say it's proteins which are present, that are still there, and the immune system can still recognize कि अच्छा ये तो वही protein है. They get stimulated and the job still gets done. But because the virus is dead, it's inactivated, it will not cause the disease. Okay, so this is a method which was used for Covaxin. So 2 and 3 are correct. Answer is B. Then you have DNA barcoding. Okay. See, DNA, I told you, very large, very extensive molecule. Sequencing is very difficult. We have more than a billion, like some 3.2 billion nitrogenous base pairs. So if we start counting that, it becomes a very complex process. You need supercomputers to analyze that data. You cannot do that every time. So what we have done is, we have identified some parts which are unique for a species. Okay, all species are different, right? Like Homo sapiens are different from, say, the previous Homo species, or within the group of cats, different cat species have some unique point. That unique point is definitely in their DNA. So what we do is, we create a data just of that unique point. You understand the concept of a barcode, which you use to make payments, right? So sabzi wale ke paas laga hota hai na barcode, which you are supposed to scan, and once you scan, all his information of his bank account, his name, everything comes. Then you will put the money in that, and with that he'll receive the money. All his bank details are automatically coming into your system with just that barcode which you are scanning. Same way, we can create barcodes, or we can store barcodes for different species. What we do is, we find out that out of the whole DNA, which part of the DNA is unique for a particular species. Okay, that part we pick and we store that information. That every time we get that sequence, we'll know that, okay, this sequence is for this particular species. How that helps us is, it will help us identify a particular species. Like, look at this. This is a very good depiction of barcodes. So there is an unknown species. Okay, suppose somebody is doing some research in Western Ghats and they've identified some species, and by looking at the species, they cannot exactly distinguish that which type of, this is an insect, which type of insect it is. So they take out its DNA and say, for this insect, we know that for insects, this part of the DNA is unique. Okay, whichever part is there. For different organisms, there can be different segments of DNA which will be unique for that species. So insects mein jo bhi part tha, which can act as a barcode, we take that. We only sequence that. We don't need to sequence the whole DNA. We just take the barcode, I sequence the barcode, then we put it into the barcode database which is available already, and we see if there is a match. If there is a species already there with the same DNA sequence, and then it is identified that this is that particular species. So Sorry? ये तो जहाँ से मैंने चोरी किया है, कॉपी किया वहाँ का है, right? Please don't read this. I'm not supposed to do that legally, okay? All right. So coming back to the question. All right. DNA barcode. Let's see what it is saying. Consider the following statements. Which of the statements given above are correct? Okay. So DNA barcode can be a tool to assess the age of a plant or an animal. So your DNA will not change with age. Okay. When you were a zygote to when you are 100 years old, DNA is the same. So there can be no information of age which you can get from any DNA. Okay. So even if you don't know what is a barcode, even then you should know that this will not give information on age. Okay. So 1 is absolutely not correct. Then distinguish among species that look alike. Yes, this can happen because DNA is unique for every species, and that means your answer will be D. Okay. Identify undesirable animals or plant materials in processed foods. Okay, so suppose you have some food item and there's some bacteria growing and you want to know which bacteria this is. You can take that bacteria, you can identify its barcode, and then you can find out kaun si species ka bacteria hai which is growing here. Okay, so that will also work. Answer will be D. Like this DNA barcode, there is another possibility of a microsatellite DNA. There are some sequences in DNA of all organisms which are repeated. Short tandem repeats. Repeats as in जैसे कि एक sequence है AGT. Suppose this AGT is now repeated 100 times. Okay. This repeated sequence becomes a short tandem repeat or a satellite DNA. Again, if we have to sequence DNA, sequencing the whole DNA is always a problem. So, ऐसे जो parts हैं which are repeated, which are repetitive, they are very easy to identify. So, पहचानने के लिए they can be really helpful. So, that's the job of satellite DNA. That when you don't want to identify the whole organism, you can just identify a small piece which can be a satellite DNA. Like you know about DNA test, DNA fingerprinting? Yes? In that the complete DNA is not identified, it's not sequenced. You just look for some satellite segments, some parts which you know are unique for an organism which will match with the parents, only those you can sequence and you'll get your information. Now let's see what it is saying. Studying the evolutionary relationship among different species of fauna, stimulating stem cells to transform into diverse functional tissues, promoting clonal propagation of horticulture plants, assessing the efficacy of drugs by conducting series of drug trials. See, अगर आप microsatellite भूल जाओ, right? If you just forget this, if you just think about DNA, okay, if you understand what is DNA, what is genetic material, how it is working, then also some options I think you can easily eliminate, especially this last one, efficacy of drugs and drug trials in population. Right, what does that have to do with working of DNA? Then horticulture plants. Clone means a copy of horticulture plants. There is no genetic technology going into this. And stem cells into diverse functional unit, again, that cannot be the case. It is going to be A. Studying the evolutionary relationship among species. Like I told you, how much DNA is common, how much sequence, how many microsatellites match. That will indicate a closer relationship in evolution. Okay. Such questions with a little bit of effort, with a little bit of, more focus while reading the question, you will be able to attempt. If you have, again, the concept, that will be important. If you've learned this part, then again, of course, they'll not give you an exact statement, but conceptually this will work. This was a question which came last time, and this again was a question which was discussed very widely, because this nobody knew what it was. Okay, no idea at all. So such questions will also be there. This, no current affairs, no news, no static book, I think probably no document in India is talking about it. And this question was asked. Question is aerial metagenomics. So of course now everybody is read about aerial metagenomics, so I'll also be able to tell you, but जब question आया था तब आता नहीं था किसी को भी. What this means is, metagenomics means when you take a collective sample of DNA. Okay, like you take a water sample and all the organisms in water, you extract DNA from all the living cells present in water. That's metagenomics. Aerial metagenomics means when you take that sample from air. Okay, you take an air sample from a place and you check all living organisms in that. आप सबका सीक्वेंस करोगे, कौन सा बैक्टीरिया है, कितने बैक्टीरिया है, किस किस टाइप के बैक्टीरिया है, वायरस है, जो जो ऑर्गेनिजम्स है, when you check all of that, that is aerial metagenomics. So collecting DNA samples from air in a habitat at one go. That is aerial metagenomics. Understanding genetic makeup of avian species using airborne devices to collect blood samples. Look at this option. How stupid is that, right? See, using airborne devices to collect blood samples from moving animals. Think of it, you know, an elephant is going in the forest and a drone is coming to collect sample of blood for some testing, right? कुछ भी. So इस तरह से, again, if you are not immediately annoyed by metagenomics and aerial, maybe this you could have eliminated. Then sending drones to inaccessible areas to collect plant and animal samples from land surfaces and water bodies. Again, answer is A. Okay. It's a little better if we leave such questions, right? Last part, I think last two questions. Drug resistance, very very extremely important. Mains में भी आ चुका है, prelims में भी आ चुका है. I told you about the basic concept that we take medicines for different pathogens. When those medicines don't work, we say pathogen is resistant to the medicine. Okay. This is a very conceptual question. Which of the following are the reasons for occurrence of multi-drug resistance in microbial pathogens? So they're asking you how these pathogens are becoming resistant. One of the most basic cause of resistance is improper use. Okay, improper use means, like suppose I'm supposed to take a medicine, I'm supposed to take an antibiotic, and the doctor asked me, please take it for 5 days. Okay, I did not take it for 5 days. Okay, I took it for 3 days, then I left it. Like this is a common trend in our country, I think all over the world also, but yes. So I have taken less dose than what I was supposed to take. The pathogen in my body, the bacteria, were now underexposed. Another situation can be that every time I have some kind of paranoid problem, every time I feel like, you know, I might get sick, I start taking medicine, antibiotic, which I don't need. So I have an overdosage of that. So anywhere where there is underexposure or there is overexposure of a particular medicine, the pathogen can find some loophole. Okay, think of a very simple example. An analogy where, you know, if your parents keep scolding you all the time, right? So शुरू में बुरा लगता है, फिर थोड़ा कम बुरा लगता है, फिर बुरा नहीं लगता है, right? Same thing, same concept with pathogens, right? If you have an overexposure of something, it might not work. Okay, and this is one of the biggest reasons for having a very large incidence of these pathogens becoming resistant to the disease. And the challenge is that there is no way to go back to it. Once a pathogen, bacteria, fungus, virus, anything has become resistant to a medicine, that medicine will never work. Your only option is to change the medicine. Okay, and there is a possibility that more than one medicine also stops working. So there can be like a pathogen who will be a super pathogen where no medicines work. Okay, there can be a situation, there'll come a point when that. Now in this, they have given you causes of occurrence of this resistance. Genetic predisposition of some people, then taking incorrect dosage of antibiotics to cure diseases. This was a very common statement. I feel this everybody should identify. So 2 should definitely be in your answer. The next option is using antibiotics in livestock farming. How this works is, suppose you are taking an antibiotic, cow is also taking same antibiotic. Okay, then the cow is giving milk. Milk mein aa gaya antibiotic. Then you drink that milk. In your body, more antibiotics. Okay, so overexposure has happened. It can happen through food products from animal sources. Milk, eggs, meat, all of that can be creating this problem. There is also a possibility of this kind of contamination happening from water. Uh, there's a pharmaceutical company who's preparing these medicines, and whatever is left, they're throwing in the water body. Then people are drinking water, again, they are improperly exposed to that medicine. That can also cause resistance. But this third statement, using antibiotics in livestock farming, that means in animals, this can also be a reason. I think these 2 statements you should be able to understand normally. The first option is automatically eliminated. However, I'll tell you, first option says genetic predisposition of some people. It means that there can be something in the genetic material of a person because of which resistance is happening. See, resistance, that is a property of the pathogen. Bacteria has become super bacteria. Virus has become super virus. Fungus has become super fungus. Okay. It has nothing to do with your DNA or my DNA. Because that particular bacteria will not respond to that medicine, चाहे वो मेरी body में है, चाहे किसी और की body में है, even if I take it to the lab, its response will be the same. Once it knows how to give a medicine, that will work like that. Okay, so it is not dependent on the condition of the person. So this cannot be a reason why pathogen will change form. Okay. Last option, multiple chronic diseases. When you talk about this word chronic diseases, these are the non-communicable diseases. Do you know anything about non-communicable diseases? Can you tell me any example of a non-communicable disease? Diabetes. Okay. So any disease जहाँ पे pathogen involved नहीं है, diabetes, asthma, high blood pressure, kidney disease, liver disease, any such disease, that will be a chronic disease. Chronic means the disease is less severe and it is long term. Okay. And a lot of people have more than one chronic disease. Like in old age, somebody will have diabetes also, then asthma also, then maybe some heart problem is also there, multiple diseases. But these diseases, when a person has, this also does not impact the virus or bacteria or fungus. If there is a bacteria coming in your body which is not responding to a particular medicine, just because you have diabetes, the bacteria will not change its mood. Okay. It has a property which it has acquired, and it is not dependent on any other disease which you have. So this also cannot be the answer. The answer is going to be B, 2 and 3 only. Okay. Immunity, if you talk about immunity, yes, आपके genetic material आपका immunity decide करता है. What kind of diseases you have, that can also be linked to your immunity, right? But here, this is talking about the pathogen. So you have to think about properties of the pathogen, right? Okay. This is a diagram to show what is antibiotic resistance. ये हैं bacteria, normal bacteria. To kill them, you have an antibiotic. If bacteria changes form, it becomes resistant bacteria, then the antibiotic will not work. फिर जितनी मर्जी antibiotic खाओ, जितने मर्जी dose में खाओ, it will not work. This, when the medicine does not work, your only option is to change the medicine. And this idea is antibiotic resistance. Okay, I believe this was the last question. Okay. Any questions you want to ask before we can end? My summary, my last note would be, current affairs will give you a very broad base on what all topics you should study. And whenever you are identifying that what needs to be studied, current affairs are going to be helpful in that. Newspaper is a very good source. In your newspaper, even if you are to the depth of every topic, prioritize on topics which come in editorials, because that automatically reflects that they are more important. They are a part of a national question being discussed. So there you must know. And on your science pages or anywhere, even if you're not going into the specific research, you should understand what it is talking about. What is the theme? What is the issue? What is the basic concept here? If you have to do a quick search for that, do that. But make sure that terms you identify. And a basic groundwork of your NCRTs goes a very long way in understanding the questions. If you saw these questions, I'm sure you have this idea that very few of these questions were purely factual. They needed you to understand things. Only then you can attempt the questions. Keep your strategy simple. Revision of concepts multiple times is what is going to be the main thing. Questions are helpful in understanding what all themes on areas you need to identify. There can be repetition of themes also, like viruses or diseases or vaccines. Prominently, you'll see them a lot more number of times in future also. So identify that and keep revising. Previous year questions hain, or you've given any test, anything which you've already done, that should be done multiple times. And when you are starting off, please start with class standard, 6th standard NCRT. Okay. I wish you all the best. Thank you.